the first 5, and then we'll take a short break, and then we'll go through the second 5. But before we get started tonight, Dr. Hogarth, I think you have a presentation.
Thank you. I'm Dr. Michael Hogarth. I teach field zoology. I know all of you know me, but maybe not those guys. And I actually present the most coveted student award here at Stone Laboratory because it's the only student award here at Stone Laboratory. And until actually just a few days ago, I did not have the credibility to do that. I now have 3 creds. Maddest Collector Award right here, and my award is the Mad Collector Award. This will be the last year this award has been given out. It's actually been given out every year for the last 19 years. The first year it wasn't given out. It was given to that guy. He never came forward to get this thing. I'm going to give him the whole plaque here after this. And he can keep it for as long as he wants to. And so we do have 2 Mad Collectors here in attendance today, Dr. Candy and our illustrious Matt Thomas, the Mad Collector. And so this is the award. And I'm thinking a little bit outside the box this year. I actually have 2 Mad Collectors this year. And so it has a lot to do with symmetry, but and there will be years and years of study to see how much symmetry actually goes into this. In 2005, I also had Mad Collectors, but I engraved these so that they would be on separate so that I could have this symmetry going on. Okay, anyway, so I have 2 Mad Collectors. One, I'll just tell you who they are so we can get on with this. And these 2 guys can come forward if they would like to. Abby and Blair, would the 2 of you come forward? My 2 Mad Collectors for 2014. Abby is the insane collector, mad in that way. This is the quiet, sort of the serial killer kind of thing. And so, between the 2 of them, they would make one really massive, keep your children out of the road kind of Mad Collector. Here, you 2 hold this.
Keep your... Watch for this on Facebook.
Watch for it on Facebook. And if I could have that.
Thank you.
I've collected them all.
Very good.
I'm done now, thank you.
That would be sweet.
And hey, Mike, before you leave, we
Oh, I get to leave?
No, you have to stay this time. No, just kidding.
Yeah, right.
No, seriously, I think we all have loved kidding around with Dr. Hogarth. He's done a tremendous job up here for 20 years. I can't thank you enough for that. We've got a couple awards that we'd like to recognize you with tonight. And Chris, why don't you show? Where is Chris? Chris, why don't you show him the show it?
You're giving permission to be there.
Show him the first one.
Yeah. What? So this has been ordered for you. So it's it's an all wooden Great Lakes clock. It was purchased for you, and it's not here yet.
I've heard that before, Chris. How was your office to pick that up?
Yes. And there's a car that's for you too, and that's no, Lindsay Manzo's actually in Annapolis picking it up.
Okay.
So she's got it.
Great.
We'll
Thank
get it
you.
to you as soon as we can. Thank you very much for all your time.
And then this other one, I think you're aware that we have a number of awards that we like to recognize outstanding people with, and we wanted to recognize you, Mike, as with our Distinguished Service Award. And this is the Stone Laboratory Distinguished Service Award presented to Dr. Michael Hogarth in recognition of your dedication, commitment, and outstanding service to Stone Laboratory as the instructor of field zoology for 20 years. You are the creator of the Mad Collector Award and the person who has won our Outstanding Visiting Professor Award more than any other faculty member. You're a shining example of what a faculty member should be. You make science exciting and fun, and all of our students, whether they've been in your class or not, we've all benefited. Where am I at on this? benefited, thanks, Mike. He's been with me too long. Benefited from your presence on the island. We really all owe you a sincere debt of gratitude. Dated July 24th, and thank you very much, Mike.
Thank you, Jeff.
You hold on to that. And just seat right there, maybe.
It's July 24th of 2018, though, so you don't get it for another 4 years.
Rosie's running away from me.
Off the rock.
Off the rock.
That's as good as I'm gonna get with you two.
Thank you very much.
You want this?
And if you, we won't move this because we have our camera on it.
We're not gonna move it?
Oh, yeah, we will. All right. The listing over here, and there's Michael A. Hogarth, right up there.
Well, thank you.
Okay.
The clock's only this big.
You know, if you ever change your mind, this is because President of Ohio Academy of Science now, I believe, and so when you're done doing that, you know, we'll have you back.
Okay, maybe.
This is I think a real important night. I love these REU presentations. The group of students tonight take us over 100. This will take us to 103 since we started this program back in about '05. They're all in outstanding places. We just saw information on Hillary flying back on an Army transport from Greenland, and Kristen, who do we have down in Costa Rica right now? Kelsey?
Kelsey's in Peru, actually.
Oh, in Peru now?
Yeah.
Okay, she had been in Costa Rica,
She
right?
was in Costa Rica for a little while, now she's in Peru.
Jeez. Okay, so the students are all over, a lot of them in graduate school, and we hope that all of you go on to that. For all of you that are here, for all the students that are in here, you're just about done. You'll finish up on Saturday. This is a real good time to also make sure you have email addresses and things of all the friends you've made up here, because the people you've encountered up here are likely to be, your friends for the rest of your life. These are colleagues, people that you've done stuff with. You've shared experiences with them, and you have a lot of shared interests. The people that I was here with as a graduate student are still my friends today, and you'll find that is the same thing with you guys. You can all join the Buckeye Fossil, our student group of the Friends of Stone Laboratory. Get involved with that if you're interested. We have open houses early September you can come back for. We have a work weekend in the spring, a work weekend that you can come up in the fall and still come back up here. And you'll find that I'm betting right now that there are a number of you that just can't wait for Saturday to come. You want to get back and put your feet on the mainland. And honestly, when I left the first time, that's exactly the way it was for me. And you'll be back about a week and you'll start missing it. You just want to get back close to the water. I hope you feel that way. And we want to see you back here. Also, think about your faculty members that are up here, the people that you've worked with. If you were working for us, check with people, see if they would be willing to serve as a reference for you as you try to get into graduate school. The students that are up here first time that were not REUs, if you want it, you've seen this experience now, if you'd like to come back next year and try to be an REU, or come back next year and try to work for us, it'd be a good time to talk with Kevin, talk to Matt, talk with Chris, talk with Justin, and see what kind of opportunities might be there. We really want to see all of you succeed. The bottom line is we hope that this experience has been really good for you. We hope that you've benefited from it. And as you go on with your careers, we hope that you'll feel free to come back and talk with us if there are things that we could do to help you get to the next level or wherever you're planning on going. With that, I'm going to stop, and Justin or Chris, Justin, are you going to explain how we're going to do this tonight? Very good. I have somebody's, I've grabbed somebody's cell phone. Anybody missing a really nice iPad or iPhone?
so we're going to do this in conference format, where there's going to be a set schedule. And in a conference, normally when you go, there's sessions. And we're going to have two sessions. So the first session is going to be in limnology. So the limnology students will go, then we'll have a short break, then everyone that's studying vertebrates will go. And then we're kind of within all of it, we're kind of going in the order of the food web. Trying to, at least. So our first presentation, Doug will introduce the speaker, and we're going to try to keep it at 15 minutes. I'm going to have a timer. At 12 minutes, I'm going to stand up so you can see me, and then at 15 minutes, I'm going to, hopefully you don't go more than 15, but.
We have this.
Something bad will happen.
Yes. Okay, I do have a couple of announcements to make. I'm on 3 boards right now. One, the Ohio Academy of Science, the other, the International Association for Great Lakes Research, and the Ohio Lake Management Society. All 3 entities have conferences every year, and the presentations that we're going to see tonight are appropriate for all 3. I'll give a special plug to Ohio Lake Management Society, because they're trying to get more and more students to present, and they only have a few every year, and they give out prizes for best presentation. It's too late for this year's, which is in November, but maybe the next November, so be thinking about that.
And Doug, are you also the president?
President of IAGLR, which is in Vermont this year, but it's going to be closer to us probably than the year after. The best news, though, is the Ohio Academy of Science meeting is in faraway Bexley, Ohio. So that should be really close for all the Ohio State students. So hopefully some of you will submit your work and present in the spring. That's enough for the promos. We will start now with the first talk. My student is Tiffany Atkinson. She is a senior honors student in the School of Natural Resources at Ohio State, specializing in water with the overall major of environmental science. She's from West Salem, Ohio, which I've actually driven through a couple times when I worked at Ashland and got gas. I don't know what else. I saw some Amish buggies and about, I don't know, maybe I saw everything. But I did see the fireworks store, which I know they have a fancy fireworks. The other thing about Tiffany is I'll never forget the first day I met her. I had gotten new glasses in Columbus, and I walk into Cotman Hall, and you know when you get new glasses, I couldn't see anything. I'm like, how am I ever going to find this person? But I sat down, she found me. So that's why I was squinting and looking. You were still there. But the last 5 weeks, she's worked on dissolved oxygen dynamics in Lake Erie's Sandusky Subbasin, and she's going to be putting into context some data that we've been collecting for the last 10 years now. Tiffany.
Okay, so as Dr. Kane just told you, I'm Tiffany Atkinson, and my project is dissolved oxygen dynamics in Lake Erie Sandusky Subbasin, and it's a 10-year analysis. Okay, so eutrophication is an excessive production and accumulation of algae and aquatic plants in a body of water due to increased inputs of nutrients, and phosphorus and nitrogen are usually what's attributed to this eutrophication. So in the 1960s and 1970s, Lake Erie had very large amounts of phosphorus entering the water, and this was due to anthropogenic sources. It was Lake Erie was at that point considered a very eutrophic system. So Dr. Seuss at the time in 1971 actually even put in his book The Lorax a little line about Lake Erie, and it said, "You're glumping the pond where the humming fish hum. No more can they hum, for their gills are all gummed. So I'm sending them off, oh, their future is dreary. They'll walk on their fins and get woefully weary, in search of some water that isn't so smeary. I hear things are just as bad up in Lake Erie." And so that was in 1971 when the system was pretty eutrophic. In the 1980s, Ohio Sea Grant asked them to take it out of the book, so in the later versions, it's actually not in the LORAC. So in 1972, the Great Lakes Water Quality Agreement was passed, and this was between the United States and Canada, and it reduced phosphorus loading by quite a bit, and Lake Erie's health improved. So in this figure, you can see that the phytoplankton biomass significantly decreased between 1970 and 1985, and this is the Central Basin and the Western Basin. So then in the mid-1990s, the phytoplankton biomass started to increase again significantly. So eutrophication reemerged in Lake Erie, and eutrophication is thought to be a large portion of the cause of hypoxia, although this is hypoxia is not fully understood, so there's a lot of dynamics that go into it. Lake Erie has been experiencing hypoxia again since 1997, even though total phosphorus loads are well below regulation. This photograph here is just a picture of microcystis in the lake, and then the figure down at the bottom shows the intensity of microcystis blooms, which have been increasing in the lake. So during the summer, the water in a lake stratifies, so it separates into different layers because of the difference in density and temperature. So in this figure right here, you can see that there's three layers: the epilimnion at the top, the metalimnion in the middle, and then the hypolimnion at the bottom. And the hypolimnion is what I was focusing on in the study. So the hypolimnion becomes isolated, and dissolved oxygen exchange between the layers is greatly reduced. And in the bottom layer, in the hypolimnion, there's a lot of respiration from organisms in the sediment. So oxygen is depleted very easily in the hypolimnion. So this is a figure that I made. The blue is the temperature curve, and the red is the dissolved oxygen curve. And you can see right here is where the hypolimnion starts. So you can see that there's a decrease in dissolved oxygen when you hit the hypolimnion. So hypoxia is considered dissolved oxygen levels below 2 milligrams per liter, but it also can be defined as 4 milligrams per liter. It just depends on which journal you're looking at. Hypoxia can lead to declines in population and extirpation of aquatic species in the lake, which is why it's so important that we need to study and understand what's going on with it. So this study focuses on the dynamics of the dissolved oxygen consumption rates of Lake Erie associated with the thickness and the depth of the hypolimnion over a 10-year period from 2005 when the program started until this year, 2014. And it's focusing on two different sites in the Sandusky subbasin. So these are the sites. We are up here. So this is the Sandusky subbasin right here. This is called East. It's the East Sandusky subbasin site. And then this is called Stock. It's the Sandusky subbasin offshore site. So for data collection, Captain Craig Sinclair would drive us out to 6 different sites. I only focused on 2 in this study because those were the only 2 sites that we had data for consistently over the last 10 years. We do have data for all of them. So when we would go out, the first thing we would do is we would take a Secchi disc reading. We would always bet to see who would get the closest to see how clear the water was. So this is a Secchi disc right here. And you just lower it down until you can't see it anymore, and then pull it up, see how far down it went. So what I'm holding here in this picture is my baby. This is what I used to get all of our data. You just lower it down into the water at half-meter increments. It would record dissolved oxygen concentrations, temperature, chlorophyll A, and phycocyanin were the ones that I was looking at. So then after that, I compiled all 10 years of data, which took a really long time. So I had a bunch of different Excel sheets, and I compiled everything into one Excel spreadsheet. I examined the last week in June and the first 2 weeks in July for all 10 years because those were the dates that I consistently had data for. So for each day, I made a chart, and it shows the temperature curve, the dissolved oxygen curve, and the chlorophyll A curve, or line. So then we did some data analysis, and for the I would delineate the temperature curve, which is shown in this picture. The black lines are how I delineated it. So the top line here is where the epilimnion stops, and then the bottom line here is where the hypolimnion starts. So that's how I calculated the thickness and the depth of the hypolimnion. So after getting those measurements, I could calculate the mean hypolimnetic oxygen consumption rate. And then I plotted them on a scatter plot, and we ran linear regressions, and magic happens and you get statistics. How I feel about them, at least. So the mean hypolimnetic oxygen consumption rate is in milligrams of dissolved oxygen per liter per day. So what we did was we took the oxygen concentration of the last week in June and subtracted the oxygen concentration in the hypolimnion from the last day or the second week in July, and then divided by the number of days that passed between the two measurements. So my results, this chart at the bottom has both soft and east as one data series, plotted as one data series. What we're showing is the hypolimnion thickness on the x-axis, which is this right here. This little red guy shows where we're looking at the thickness. So we were looking to see if the hypolimnion thickness had any effect on the oxygen consumption rate in the hypolimnion, and we had significant data that the thickness does affect it. So what it's showing is that as the hypolimnion thickness increases, so does the oxygen consumption rate. And our original hypothesis was actually that it would be the opposite of that. So you would, instead of seeing a line go up like this, we would see one go down like that. So it was the opposite of what we thought. So then when you separate out east and south as two different data series, it actually changes the dynamics a lot, which is not significant data this way. So the yellow dots down here are 2014 data, and in this one it's really interesting because they actually overlap for both sites. So then I also plotted year versus the hypolimnetic oxygen consumption rate, and there was not a significant correlation in that graph. And then I also did the hypolimnion depth. So what we're looking at in this graph is this point right here. So this one is about 10 meters. So we were looking at the depth of the hypolimnion in the water column. So what we saw is that as the depth of the hypolimnion increased, the rate of oxygen consumption in the hypolimnion decreased, and that was a significant correlation. Then when we separate them out as two different sites, it actually was not significant anymore. So then these last graphs just show the second week in July. We saw that there was still a lot of dissolved oxygen in the water this year in the hypolimnion. So I decided to do to see what the concentrations looked like over time the second week of July. This is the mean hypolimnetic oxygen concentration, and you can see that 2014 has much higher oxygen concentration still. It's about 2 milligrams per liter higher than the highest year at soft, and then it's about double that in east, which I thought was really interesting. So this paper was actually an aerial hypolimnetic study, and the aerial hypolimnetic mineralization, which is on the y-axis here, is the total aerial hypolimnetic oxygen depletion, including the formation of reduced substances. So it's not just the bacterial respiration, it also includes redox reactions. And then the bottom is the mean hypolimnetic thickness. So at first we thought that we could compare our study to this one, but it was pointed out to us that the aerial oxygen consumption is different than volumetric, and we were doing a volumetric study. But this does show the same trend that ours shows, so it was interesting to look at. This one is also one of their figures that shows that the smaller the thickness, the smaller the oxygen depletion rate. So this one is on the same scale as ours. It's milligrams per liter per day, and this study is in the central basin of Lake Erie. So it shows that the complete opposite of what ours showed. As the hypolimnian thickness increases, the rate of oxygen consumption decreases. So it's really interesting to look at ours because it's the complete opposite. So something that could have caused this is that the central basin, the Sandusky subbasin morphologically is the same as the central basin, but as far as inputs into the water, it's more like the western basin because there's a lot of agricultural runoff. So that could be where some of our differences are coming from. So then these last two charts that I want to show you are about ice coverage, and this one shows the weekly ice coverage for between 2005 and 2014, and you can see that in 2014, so this is ice coverage after, it's between April and June, so you can see that there's way more ice coverage in 2014 than there is any of the other years. This black line, just disregard because that one says no data. I couldn't get it to go away. So then this one shows the total accumulated ice coverage, and this is from December until July. And you can see here that the 2013 and 14 season definitely has more than the other years. So that was just something that we decided to look at because we were interested about why there was so high of concentrations of oxygen this year. So it was still just kind of something to look at, like maybe this is the cause. So in general, more research definitely needs to be done because hypoxia is still really poorly understood, and there's so many different dynamics that go into it. So it's really important to study it. And another thing, although this is a 10-year study, the temporal scale for each year is only about 14 days. So there could definitely be some improvements with the temporal scale. And that's it. These are my acknowledgments from the people who made it possible for me to do all this. So, any questions? Yeah.
So I was wondering, do you think that the storm events could have played some sort of role in the high oxygen content this year?
Yeah, so when there's a really big storm event, how I showed you the temperature delineation, it will actually mix the whole system, and all of the oxygen will mix up into the top and the bottom of the lake. So that definitely could have something to do with it. The weeks that we went out and sampled, we didn't have any mixing, so we would have followed a straight line instead of like a curved line. But it really switches back to being stratified pretty fast, so it could happen in between.
Yeah. You showed your ice cover, you're, I guess, showing that they've possibly correlated with your consumption rates. Do you have ideas as to why, or like just thrown out?
Like why it would be different?
Why that might be a reason?
So it was kind of just like a, hey, maybe this could be why, but we didn't really have the resources or the time to compare them. So really it's just a whole other study in itself of why that could be affecting it, honestly.
Tiffany, the study that yours differed from
Yeah.
was done in the central basin, whereas yours was in the sub-basin.
Mm-hmm.
What kind of, I don't know if you remember or not, the minimum and sort of maximum, the range of thicknesses for the hypolimnian in the central basin versus the range of thicknesses in your basin?
You know, I actually didn't look at that in their paper. Probably would have been a good thing to look at, but the range for ours was anywhere between 1/2 of a meter and like 4.5 to 5 meters.
Yeah.
So there was a big range.
Big range, and check whether their minimum was less than yours. And then also give some thought to if you might have any diffusion of oxygen going on. The smaller you make, the thinner you make your hypolimnian. When you have if there's any diffusion across the thermocline or across the metalimnian, the influence of the diffusion is greater if if it's thinner. It may have absolutely no impact, but it might help you understand why you would see that. And I would think that you might have a much larger minimum out in the central basin itself, so the size of it.
Right.
Yeah. That's very good, though.
Any questions?
So our next presenter is Laura Smith, and Laura, why don't you go take your rightful place. I don't have quite the biography of Laura that Doug did. I apologize. Laura is a senior. She's going to be graduating from the Ohio State University, and she has been working with me this summer on her REU project, and it's a very exciting one. She was asking me the other day if I was happy with the project. I said, yeah, this is great. We just got this brand new machine, and you were the first one to use it. It gave me an excuse to go back to Kent and trial this new machine. So it is an exciting project, and I hope you'll enjoy it.
Okay, so I guess I don't have to introduce myself again, because if you haven't gotten it by now, you're not listening to me anyway. But my name is Laura Smith, and I'm studying the ecosystem nitrogen balance and the applicability of the membrane inlet mass spectrometry for measuring nitrogen dynamics in Lake Erie. And obviously my advisor is Dr. Darren Beatty. Okay, so the nitrogen cycle is actually a lot of different fluxes and pools going on all at once, but we're just focusing on two of them, which is N fixation and denitrification. We really want to know which one was dominant in the area around here, and how much N fixation was contributing to the pool of reactive nitrogen in the water. So that first equation is for N fixation, and really the important part is that it's taking dissolved gaseous nitrogen, which is an inorganic and unavailable form, and converting it into ammonia, which can be used by organisms. And also, if you notice the 8 electrons there, that means it's a very energetically expensive reaction, and it's not going to go unless you have the right conditions to favor it. Denitrification is, in some ways, like the inverse. It takes NO3, which is a form used by organisms, and puts it back into an unavailable N2. So if we had no biological processes going on and we just had nitrogen in the atmosphere and nitrogen in the water, they should be in equilibrium, and we should see the same concentrations throughout. But you end up with N fixation, which can pull down N2 and put it into the pool of reactive nitrogen, which is their various forms. NO3 is one form, NH4 is another, and then there's also other forms of organic N, like amine groups that are attached to other organic molecules. And then denitrification will bring that back up back into N2. So if we see a lot of N fixation in the environment, we'll expect our dissolved concentration of N2 to be lower, so an undersaturation in the water, because it'll be pulling it down faster than it can be pushing it back up. If we see a lot of denitrification, we'll see the opposite. We'll see an oversaturation of N2 in the water for the same reason, the arrows. So the reactive nitrogen pool really shouldn't change that much because there are more things affecting this. It's not quite this simple, but the arrows are nice. So we already said it's very energetically expensive, and this is because it has to be catalyzed by nitrogenase. So this is very difficult to synthesize. It takes a lot of energy on the organism's part. So if there's any available reactive nitrogen in the water already, such as like if it's just floating around a whole bunch of ammonia or ammonium ions, that's actually going to suppress the synthesis of nitrogenase. And so you're not going to see any N fixation because it's not necessary. So we have two different types of N fixation that we're looking at. The first is autotrophic, and that's actually light dependent. That is the one that would be happening from the cyanobacteria out in the lake in the photic zone. And some examples of this are Anabeta and Aphanizomenon. Both of these are N-fixing cyanobacteria that we hear about all the time. They're implicated in algae blooms. Another form that I really wasn't expecting to see much of here is heterotrophic. It can happen in the benthic area, so where there shouldn't be light penetrating. And so it's not light dependent. It can happen in the dark. It's not really as important for lakes, which is again why I didn't really expect to see much of it. So, why everyone should care, just in case nitrogen doesn't excite you just by being nitrogen. it's very important in the role of the production of algae toxins because it's a key component of those toxins. So if you don't have enough nitrogen, you can't make the toxin that uses it. Some cyanobacteria are capable of N fixation, but some are not. So a lot of the ones that you hear about, like microcystis and planktothrips, those actually don't fix N. So if you have an algae bloom, and it's composed of both N-fixing and non-N-fixing cyanobacteria, and you decide the way to mitigate that algae bloom is to reduce the nitrogen inputs into the lake from fertilizers or other anthropogenic inputs, you might actually just be favoring the N-fixers. And the end result of that is you will have spent a lot of money and made farmers very angry without actually solving the problem, because you'll still get an algae bloom at the end of that. So understanding the N cycling of the lake can help us make more productive management decisions. So I sampled 7 different sites. We hit SCB twice. These were over the course of 2 days. So for the first experiment, sorry, I skipped a slide. Okay, so my hypotheses were that if we have reactive nitrogen concentrations that are low, then we expect N fixation to be happening. And if the rates of N fixation or denitrification are large enough, our membrane inlet mass spec should be able to detect nitrogen undersaturation or supersaturation in our environmental samples. Okay, so we had 7 different sampling sites. For the first experiment, or relating to the first hypothesis, we had incubation experiments at sites 2 and SCB. I had the lake samples are just kind of like controls, and then 2 different treatments, and I had 5 replications of each one. For our N2 saturation experience relating to the second hypothesis, we sampled at sites E, 3, 4, 5, 6, and SCB again on the second day. And as you can see, all of these sites form a transect that goes kind of from a more oligotrophic area away from the nitrogen inputs into the bay at SCB, which are very close to nutrient inputs. So if I were going to, I would expect N fixation to be at SCB if it were going to be anywhere. So I collected water from a depth of 1 meter using a Van Dorn bottle for all of these experiments because we were really looking more at the autotrophic, so we were thinking it would be light dependent, and we weren't really worried about differences in depth, so this kept everything consistent. Okay, so for my incubations, my lake or the control samples were collected and just put on ice to stop any biological processes, and this preserved the nitrogen concentration that was in the lake water at that time. For both the light and dark samples, we collected them and incubated them for about 6 hours, so any infixation that was happening, whether it be autotrophic or heterotrophic, should have continued during that time. The difference is that in the light ones, they were in a bottle permeable to light, so that would allow both types of infixation to happen, and in the dark ones, we should have only had heterotrophic. So all of our samples were analyzed using the membrane inlet mass spec, and the gases we measured were N2, O2, and argon. We really only focused on N2 and argon in this experiment, and argon here was used as just kind of a baseline to show what would be happening to the concentrations as well as physical processes only. So it's pretty inert, it's not going to react with stuff in the water, nothing's taking it up, so that should show us only physical processes and no biologics. We used an ANOVA test to detect differences in the nitrogen concentration between treatments. So if I had a difference from my initial to my light bottle, I knew that was my total N fixation value. The same is true with the initial minus dark being heterotrophic, and then if I took the heterotrophic minus the total, I knew that would give me my light sample, and that's what we were interested in. Okay, so for our saturation samples, all of these were just put on ice after collection to stop the biological processes, and then we analyzed them the same way with the membrane inlet mass spec. There were some outliers, and we removed those using Grubbs test, not just because they irritated me. Okay, so this is the machine that I used, and it's pretty exciting. It's a good machine to use for this experiment for 3 reasons. One is that it's really high precision. Another is that it minimizes the contamination between your sample and the atmosphere, because if you have a water sample and you want to know the dissolved gases in it, as soon as you open that sample, it starts to equilibrate with the atmosphere. So minimizing this as much as possible gets you the most accurate data. This is also pretty novel because we're measuring nitrogen directly, The more traditional way of doing this experiment is to measure the reduction of acetylene to ethylene, and that's using that as a proxy for measuring the amount of nitrogen that was in there. A little bit about how this works is that your sample gets drawn in through a tube that goes through a vacuum chamber, and that tube is permeable to gases. So that vacuum chamber creates a pressure gradient, and then those gases are drawn out of the sample and then analyzed by molecular mass using the mass spec. Okay, so our results, this is the nutrient analysis that our class conducted on our first day in the Sandusky Bay area. And we found that there was fairly low nitrate concentrations in the sites that were closer to the bay, which is a good start because if we have low nitrate, right, that means we don't have one of the things that might inhibit N fixation. So this is good conditions for N fixation, and if I were going to expect it to happen anywhere, it would be at either 6.5 or SCB2. Luckily, we did find evidence for it at SCB. We didn't find any significant evidence for 2, but the rate that we found at SCB was pretty high. And surprisingly, we got a lot of heterotrophic N fixation, but it was a significant value, which was very exciting. It was a little bit high when you compare it to other values reported by the literature, but it's not so high that it would be like that I would think immediately we had errors in our processing. Okay, so for our percent saturation experiment, anything that is above the line and with an asterisk or double asterisk is statistically significant and is oversaturated, so therefore it suggests denitrification. Anything below the line with an asterisk is suggestive of N fixation because it's undersaturated. And then the different asterisks just indicate different levels of alpha. So we actually did have quite a bit of significant values here, but all the differences were pretty small. If you look at the actual values on the axis, it's all within 1% difference. It was interesting to note, though, that this is the same site over 2 days, 2 consecutive days, and they are statistically significantly, or they are actually different at a p-value of 0.009, which means that it's interesting because it could be like evidence that nitrogen fixers are pulling down the amount of nitrogen in that area over the course of the day. But to get back to our differences, we decided to calculate how big we could expect the differences to be because these were all pretty small. And we did this using a mathematical equation that depended on the gas transfer velocity. Dm is mixing depth, N2 equilibrium is what concentration you would expect at the temperature and pressure at that site and day. And given an N fixation value, if you plug in all those constants, you get the N2 concentration value that you can expect to measure there. So we found that as the gas transfer velocity increased, it became harder and harder to find the deviation from the equilibrium. And this was some of the bacteria, or the cyanobacteria that we collected from SCB on day 2. As you can see on the left, that's mostly Planktothrix and Microcystis, which are both non-N fixers. But if you look really, really closely, you can find some Anabaena and some Phanosomenon, which are both N-fixing bacteria, and they both have heterosys in them, which is a specialized cell for N-fixation because it makes an anoxic environment. N-fixation can't happen in oxic conditions. So it's technically bad for the ecosystem, but I was quite excited because it supports my hypothesis. So in conclusion, my incubations provided evidence for N-fixation at SCB. My saturation samples gave significant but small differences in N2 saturation, suggesting low rates of N-fixation and denitrification. And the membrane in the mass spec can better detect nitrogen imbalances when N-fixation rates are high or the gas exchange rate is low. My acknowledgements are Dr. Darren Beatty, my whole ecosystems class for helping collect samples, Caitlin Dickinson for helping me run some of the samples, Nick Johnson for computer something, Dr. Justin Stephan for doing the nutrient analysis, the captains Craig and Art for driving us everywhere, and Ohio Sea Grant and Friends of Stone Lab for providing their money most generously.
So, the denitrification stuff, did you look into any more what type of bacteria would be doing that, and whether in Sandusky Bay or Lake Erie they found those types?
I didn't actually look into it. Denitrification also can happen in toxic environments, so I wouldn't expect it to be things that are living in the photic zone. So any of the bacteria I mentioned are all going to be in the presence of bacteria, so if they don't, and they don't, as far as I'm aware, there are not specialized cells for denitrification.
Do you remember when you were in Sandusky Bay, how big was Becky Dust? The photic zone's very small in Sandusky Bay, so one thing I think about is that even though it's shallow, there may not be getting light at the bottom.
Did you look at alternative sources of ammonia and not just from almost nitrogen, like maybe fish byproduct or some other?
When fish urinate, it's ammonia or urea, right? One of the two stand conditions. We didn't look at urea. Ammonia values were below saturate, so I wouldn't expect that to affect it. Nitrate was used. We have actually results for other forms of nitrate or nitrogen. Nitrate was the only one that's been used.
I was going to ask if you would recommend removing nitrogen from runoff.
No.
No, how come?
Because if you remove nitrogen from the runoff, you're going to favor those few little anabenas that are in there sitting ready with their heterocysts to fix nitrogen. We'll have a lake full of anabena, and it's still really toxic, slightly differently toxic than it will be otherwise, but it's a lot of economic effort for not a lot of result, possibly.
That was very good, Lodge.
Well, our next speaker is Victoria Simons. She's a junior from the Ohio State University, majoring in microbiology. And our two cameras aren't talking right now. But Victoria was up at Stone Lab before. She was an 8th grader up here, right, in one of our field trip groups. And before she came to Stone Lab this summer, she had quite a wide background. She was worked at the zoo, right, a little bit. She worked with, I believe, those chickens.
Yes, the research project studies the behavioral modifications of domestic chickens on a consciously loaded diet.
And she was off as a microbiology major. She's been working with bacteria, developing genetic library. So her background's quite broad now. She chickens, bacteria, and now she's working on algae blooms. I'm like, very good.
Hi, good evening, everyone. As Justin said, I'm Victoria Simons, and my REU project is on studying different water sampling methods on Lake Erie. And before I talk about my actual project, I want to tell you a little bit about why we collect water samples and why you should care. Water samples are primarily collected in order to determine the general health of a lake. We can analyze water samples for nutrient content as well as things like toxins, which can be harmful. The samples from Lake Erie are primarily analyzed for chlorophyll and phosphorus. We look at chlorophyll because it's the way to measure how much algae is in the water, and we look at phosphorus because it's the key limiting nutrient in algal growth. So in essence, it can help tell us how much algae could be in the water. On Lake Erie, there are many different organizations that collect these water samples. However, they all boil down to using about 4 main methods. There's a Van Dorn sampling technique, which is used by the Ohio Environmental Protection Agency. There's a twice-a-second depth reading, which is used by ODNR and USGS Sandusky, among other organizations. A water column sample, which is used by OSU through Stone Lab, the University of Toledo, and USGS out of Ann Arbor. And finally, a 0 to 2 sample, which is used by charter boats. And I'm a very visual person, so I have this visual diagram depicting all of the methods. Our first method is the Van Dorn sampling technique by the EPA. you'll notice they collect a sample from a 1 meter depth, and then a sample 1 meter from the bottom or from the thermocline, and then a third sample halfway between those 2 points. They take those 3 collections, combine them, and that's what gets analyzed for chlorophyll and phosphorus. The twice the Secchi depth reading, first you measure the Secchi depth, and then it's a sample from the surface to twice that reading. And this is done to effectively capture the photic zone of the lake, and we care about the photic zone because that's where most of our algae should be, because you need light for photosynthesis. The water column sample is a sample from the surface to about 1 meter above the sediment. And our charter boat sample, the 0 to 2 sample, is from the surface to 2 meters, irregardless of things like depth or the Secchi reading. What my project was specifically looking at is, are these 4 very different methods actually comparable to each other? Do they give comparable data? Additionally, we want to know whether the methods themselves are precise, meaning will they give similar results when they're repeated? Data collection occurred at 11 sites from Lorraine in Central Basin to the mouth of the Maumee River in Maumee Bay, and 2 to 4 of the methods were used at every site. The reason that we couldn't use all 4 methods at every site is, especially in the shallower waters, a lot of the collections became identical. For example, if our Secchi depth reading was 1 meter, then the twice the Secchi depth becomes a 0 to 2 meter sample, which is the exact same as their charter boat. However, I actually have a total of 347 data points, which 123 of those came from these 11 sites in 2014, and then I combined this with 224 samplings from 2013 to get a better idea of the overall picture. For testing the precision of replicating each method, at the first 3 sites, we did 3 replicates of each method. So instead of just collecting 1 water column sample, I would actually collect 3 separate ones. And out of every bottle, every time you do a collection, you can fill a 1 liter bottle. We did 2 filters for chlorophyll A and 2 flasks for total phosphorus. And this was done as sort of a self-check, because if you have a thoroughly mixed bottle of water and you get 2 chlorophyll filters out of it, it should have about the same amount of chlorophyll on it. So if you're getting extremely different values from those 2 filters, you know you're doing something wrong with your method. Battery might have just died. Or we froze. Did I break it? There we go.
Good.
Yep, I'll just use the arrow keys, it's fine.
It's batteries.
Alright. When we get into the laboratory, for analyzing chlorophyll, this is determined by the amount of plankton left on a filter. So we take a certain amount of lake water, we filter it using vacuum filtration onto a 0.7 micron glass fiber filter, and then we analyze it using a DMSO extraction method. Some methods, we also did an acetone extraction and compared the DMSO to this acetone method. I'll get into that a bit later. For phosphorus, we used unfiltered water samples, and it was digested with sulfuric acid and potassium persulfate, and analyzed as phosphate on the nutrient autoanalyzer. All of our samples, we ultimately compared to the water column. So we have 4 different methods, and we chose the water column and compared the other 3 methods to the water column sample. We chose to use the water column as sort of our standard because we figured it would be the most likely to catch a stratified algal bloom, since we're collecting from the surface to 1 meter above the bottom. And concentrations for each method were plotted against these water column concentrations. And now we get to the fun part, which is the colorful graph. Here, we're looking at chlorophyll first. On the x-axis, we've plotted our water column chlorophyll concentrations, and on the y-axis, we have our different comparison methods. So basically, we're comparing how much chlorophyll we got with our comparison method to the water column method's chlorophyll for each site, and the dashed line represents a perfect 1-to-1 correlation. You'll notice all of our data points are extremely close to this 1-to-1 line, meaning that we got very similar values for our comparison methods as the water column. So that's chlorophyll. Looking at total phosphorus, again, we get a very nice correlation between all of the points. They're all very close to that 1-to-1 line. Next, we're looking at how precise is each sampling method. So this is comparing those replicates to each other. Again, we did this at 3 sites and did 3 sample replicates for each method. So in total, instead of having 4 bottles for these sites, I have actually 12 bottles, with 2 filters from each bottle for chlorophyll and 2 filters from each of those for total phosphorus. So a total of 24 data points for chlorophyll and then for phosphorus. Here we did the 3 sites are off Cedar Point, Sandusky East Bay, and North Bass Island West. And each of the 3 bars, they represent either replicate 1, replicate 2, and replicate 3, and each bar is an average of the 2 filters we did from each of those bottles. And you'll notice that our biggest difference here is only about 5 micrograms of chlorophyll per liter among all of the replicates, and that's a really small difference. It doesn't start being a big difference until we start getting into the double digits. A 5 microgram difference is really not big, and most of them were smaller than that. Looking at total phosphorus, again, there was very little variation among the replicates for all the methods at all the sites. Our biggest difference was only about 0.4 micromoles per liter, which is a very small amount. What should be noted, however, is that almost all of these samples were collected in relatively clear waters. However, 2 of the data sites from 2013 were actually collected while there were algae blooms present and at sites where we had visible surface scum. When you look at those, something interesting happens. You'll notice at our mid-July Anabino bloom on the left, the charter boat sample is showing us almost twice as much algae as the other methods. And in our early October microcystis bloom on the right, the Ohio EPA method is showing almost 10 micrograms per liter less than the other method. This is concerning. It means a possibility that maybe the methods aren't as precise when we have visible surface scum. However, 2 data points do not make a story. So, future studies should look at using all of these methods at sites with visible surface scum to see if the methods continue to be comparable and precise. The last part of my study was the acetone DMSO extraction comparison. I did this for all 11 sites using just the water column method. So, all of my samples were extracted using DMSO for all of my other tests. And using DMSO, we actually placed the filter inside the test tube at 10 mL of DMSO and put it in a hot water bath just to speed up the extraction process. This acetone method we compared it to actually uses a 90% acetone solution, and then it's sonicated for 2 minutes to take the algae off the filter, all the chlorophyll off the filter. And then we're looking at the differences between the acetone and DMSO replicates. So this is my familiar 1-to-1 line here showing the DMSO chlorophyll on the x-axis and the acetone on the y. And all the data points are very close to the line, showing a strong correlation between them. However, as you'll notice, in the upper right, we have that one little lonely data point, where for DMSO, we got about 100 micrograms per liter of chlorophyll, and for the acetone, we only got 45. That's a really huge, over 50% difference between the two. That means that there's a possibility of these methods not being comparable at high concentrations. Perhaps we could fix this by sonicating a sample for longer when we have more chlorophyll on it, using the acetone method. That could be something future studies could look at. We also looked at the relative difference among the replicates. So, as I mentioned near the beginning, when we're doing the 2 filters for each method, we should get about the same amount of chlorophyll on each filter. But of course, there's still going to be some difference between them. This is looking at the mean relative difference between those 2 filters for each of the 2 methods. For acetone, we had an average about a 5% difference between the 2 filters. For DMSO, we had about a 7% difference. Those are both very, very small relative differences, and the difference between the 2 methods only about 2%. This is showing that they are very comparable to each other. It should be noted as well, however, that there are greater relative difference seen at lower concentrations. Our x-axis here is a logarithmic scale, just to give you a better idea of what the lower concentrations look like. Notice that the data points are a lot more varied, showing us greater relative difference. When we get to the higher concentrations of chlorophyll, there's a lot less relative difference. Now, I just ran through a whole lot of figures and numbers. Overall, what I want you guys to take away from this presentation is that we found that all the methods were very comparable for both chlorophyll A and total phosphorus. And this is very important because it means that the data can be shared between organizations. So whether we're looking at ODNR, comparing it to EPA, comparing it to the data from Stone Lab, even though they're using different methods for collecting these water samples, all the data that they collected for chlorophyll and phosphorus should be relatable and comparable to each other. We saw some differences during bloom events, and these should be explored in future studies to see if the methods continue to be comparable even at high surface scums. We saw differences between the acetone and DMSO methods seen at high concentrations, and that should be explored further as well. However, based on the average relative difference, the methods were very precise. And with that, I would like to thank Dr. Justin Chaffing for being a wonderful research advisor, Brianna Zellner, Karen Ortega, and Laura Smith for helping me collect my samples and also helping me in the lab with extractions and analysis, Katie Starwall for collecting all of my Central Basin samples, Friends of Stone Lab for making it possible for me to be here, Ohio Sea Grant and the University of Michigan Water Center for providing funding for the actual project, Captains Craig and Art for helping drive the boat and taking me to all of my sampling sites. Sometimes during extremely rough water conditions, there were some interesting days on the boat, I'm not going to lie, and so I really can't thank them enough for helping me out. And with that, I have plenty of time for any and all questions you guys may have. talker when I'm nervous.
So, you had 2 bloom events. Do you know how bad those blooms were in the scale of everything?
I don't know overall how large they were, but I do know that there was visible surface scum, which is the key factor we're looking at here.
Because I actually would have thought that the EPA method would be even worse and more different than, and that's why I don't know how bad, like if it's a really bad bloom, they're going to miss that whole top left.
And that's exactly what we're concerned about. We're concerned especially with the EPA method missing the surface scum. And you saw for the early October event with the microcystis bloom, it did show about 10 micrograms per liter less. But again, 2 data points don't make a story. So that's why we definitely think it should be explored further during these bloom events. Because yeah, the EPA method is our primary concern, is missing that surface scum.
Why was the charter captain sample the one that showed the greatest concentrations when you had the surface scum?
We have the surface scum? We don't know exactly. It was only one data site, but it is curious that we're showing almost twice as much. So we're not sure if maybe with the other methods, for example with the water column method, since we have overall a larger water sample, if it's diluting that surface scum more. With the EPA method, it's not taking the surface scum. With the twice the Secchi depth, I don't know the Secchi depth reading for that day, but depending on how deep that was, it could have been diluting it more. With that standard 0 to 2 sample, regardless of all conditions, I mean, we're getting a lot more of the surface scum on average, possibly.
I think your dilution thought is, that's a really good suggestion.
Yeah.
That's very good.
That's why testing more, we want to see, is it continuing to dilute it, especially with the water column samples? Is the EPA method going to miss it? Again, we can't tell up from those 2 data points, but it's enough to warrant further study, definitely.
There's a lot of people that are going to be very happy that the methods showed that were as comparable as they are. And also that they were precise.
Oh, yeah.
Sort of equally precise.
Yeah, there was very strong correlations among the data points for chlorophyll and for total phosphorus, comparing them to the water column sample. We did linear regressions, and the R-squared values were all 0.98 or greater, which, yeah. For those who know the statistics, that's crazy. So yeah, there was very strong correlation, and that's good. I was concerned about ruffling some feathers possibly if we found out maybe a certain collection method wasn't giving us wildly different results. So we'd
have been even happier if you found out that Stone Lab in Ohio State was absolutely the best method.
We did compare everything to the water column method, so yeah.
So your one really outlier data point, where you, at a very high concentration, you saw such a huge difference in the red concentration of the method. What's going on there?
Well, that's what I was thinking, is that there's so much chlorophyll in the filter. There's over 100 micrograms per liter if we're going by the DMSO method at least. We're thinking that perhaps with the acetone method, since it's using a sonication and we do it for 2 minutes, irregardless of how much chlorophyll might be on the filter, that maybe since there's more on it, it would be harder to knock it all off with only 2 minutes of sonication. And so that's something that could be explored further, that maybe if with a darker sample color, we know we need to sonicate it for longer, maybe the overall process needs to be sonicated for longer, or in order to knock all the chlorophyll off, or maybe the DMSO is somehow overestimating. So we only have one data point.
Sounds like a whole study in itself.
Yeah, exactly. That's what I'm thinking. Possible future studies.
Yeah.
I feel like a lot of times studies in general raise more questions than answers.
Those are the best studies.
Okay. All term you've heard I had half an REU, but I'm going to do the whole presentation of introduction. Not the whole, not that, the whole introduction presentation, not the whole presentation. Katie Starwalt is a junior at Ohio State in the School of Natural Resources in environmental science with a water concentration, and this is the summer of islands for her. She's been on Fiji, Australia, and now Gibraltar. So hopefully this is the best of all 3, and make sure you know, not the size of the island. So, the other thing about Katie well, a couple things. She sampled right downstream of where she lives, because she lives on the Sandusky River, and we were sampling out right in the Sandusky subbasin. and Central Basin. And the last thing, and this was the most amazing of all these things, is that she knew what songs I was singing all the time and would get the next line, even early 60s songs, which no one nowadays gets. So I have hope for the youth of the future. So, Katie.
Hi, I'm Katie Sarwell. I'm talking about nutrient limitation in the Central Basin of Lake Erie. My advisors were Dr. Justin Chaffin and Dr. Doug Kane. So a little background on Lake Erie's harmful algal blooms. Eutrophication is what happens when a lot of nutrients enter a lake. Cultural eutrophication is what happens when it's the cause of humans. So cultural eutrophication hit its highest in the 60s and 70s, and this is mostly runoff from farms, inadequate sewage treatments, and also from some detergents. So there was an initiative to reduce nutrient runoff, and also the Great Lakes Water Quality Act, or Water Quality Agreement was installed. But nonetheless, re-eutrophication started in the mid-1990s, and in 2011 we had likely the worst algal bloom in Lake Erie history, which is shown in the picture, like green. So why does it matter? Lake Erie provides drinking water for 13 million people. The tourism, travel, and sport fishing industries total $11.5 billion. And also the health of fish, aquatic animals, birds, and other animals is an important thing to take into account. Often harmful algal blooms reduce toxins that are not good for animals. Right here, you don't want to see this when you come to the lake. You want to see pretty things like this. So most of the research on algae has been done regarding phosphorus and nitrogen limitation, and a lot of the research has been done in the western basin of Lake Erie, which is over here. But there's very little known about the Central Basin, this area right here. And when I say Central Basin, I'm just including the Sandusky Subbasin, which is this area right here. One thing we do know about the Central Basin blooms is that it's Dolichospermum, previously known as Anabina. So Dolichospermum blooms in the Central Basin. Like I said, they're formerly known as Anabina, and they're a potential nitrogen-fixing genus. Previous sampling indicates that this species was in high nitrate-containing waters in the Central Basin. So the question we're asking is, do trace metals play a role? Trace metals are metals that occur in nature in very low concentrations, and they play important roles in processes of animals and plants. And so the three we're looking at are iron, molybdenum, and boron. Iron and molybdenum are needed for nitrate assimilation. So if nitrate levels are high and there's enough iron and molybdenum around, then there's going to be no end limitations. There's no nitrogen-fixing cyanobacteria like Dolichospermum. If nitrate levels are low, then there are nitrogen limitations, and they have the potential for nitrogen-fixing cyanobacteria. So this shows the nitrogen cycle and going in as N2 and coming out as ammonium. And then boron we're also looking at because it's needed for heterocyst formation. Heterocysts are these little light green bubbles right here. This is Dolichospermum. And they're the key powerhouse for making, or for fixing nitrogen. The Central Basin has these end fixers, but they don't have their heterocysts. So they can't fix their own nitrogen. They have to use the nitrogen that's in the water. So a possible explanation for this could be that boron is limiting. Without boron, they look like this. They're like dying and dead and stuff. And over here, it's really healthy and it has a nice protective envelope. So our hypotheses, low iron and molybdenum concentrations are constraining nitrate uptake and algal growth, and low boron constrains heterocyst formation. So what we did was I went out and collected 35 liters of surface water from the Central Basin of Lake Erie at sites Avon and Sauce, which you'll see in a minute. And I had 24 of these little plastic bottles. We added 1 liter of lake water, and we had a control where we added nothing, And then phosphate ammonium, phosphate and ammonium, phosphate and iron, phosphate and molybdenum, phosphate and boron, and then 3 of them got all. So they got phosphate, ammonium, iron, molybdenum, and boron. So 24 bottles total, 3 of each treatment. Then we incubated them and grew some algae. They were incubated in a light-dark cycle to match sunrise and sunset and at surface water temperature. Experiment 1 ran for 7 days before termination, and experiment 2 ran for 4 days. The reason I did that was because Avon had less initial algae in the water, and Soft had a lot more algae in the water. So we didn't want it to become a factor that it was limiting. We didn't want any other factors to become limiting. So here's Avon. After the termination of the experiment, we completed a series of bioassays, the first being chlorophyll A concentrations. So I filtered for chlorophyll using vacuum filtration. This is a little filter with chlorophyll on it. And then we extracted chlorophyll using DMSO, which is an organic solvent. We heated it up and measured using a spectrophotometer. This is what it looks like heated up, and I had to put it in all these little things and centrifuge it and measure wavelengths in a spectrophotometer. And then I also found nitrate and ammonium concentrations using the Steel Quattro flow-through analyzer. So what we did was we measured the nitrate and ammonium concentrations for the initial water sample, and then at the end of the experiment, we measured all of the nitrate and ammonium concentration for each treatment. And so what it does is it takes their water up into these little tubes, and it gives them reagents, and if that nutrient is there, it will turn a color, and then it reads it in a spectrophotometer and spits out these little peaks and stuff, and then it tells you the concentration. Very fancy. So the results from Avon. Chlorophyll A is the highest with phosphorus and boron. Down here we have all of our treatments. The black bar is the initial, and over here we have the chlorophyll A concentration in micrograms per liter. So phosphorus and boron is the highest concentration, followed by phosphorus and iron. These letters at the top basically represent whether things are statistically the same or statistically different. So statistically, phosphorus and boron and phosphorus and iron are the same, but statistically, phosphorus and boron and the all treatment are different because they have 2 different letters. So it's just a quick way that you can look at this and see what's different, what's the same. This shows the nutrient concentrations for nitrate and ammonium against all the treatments. Initially, we had a large amount of nitrate, and each time we added phosphorus, it decreased significantly, except when we added ammonium. So this one had phosphorus and ammonium, but it didn't decrease. This one had phosphorus and ammonium, but it didn't decrease. Also, ammonium concentrations were initially very low, but every time we gave them phosphate, they decreased completely. The only time they increased is when we gave them ammonium. So results for soft. Again, chlorophyll concentration on this axis and the treatments down here. Initial and control are basically the same, so there was almost no growth there, but we saw the most growth with phosphorus and ammonium, and then second highest growth was phosphorus and boron again. So nitrate depleted with phosphorus without ammonium, again, like it did in the first experiment. This was the initial concentration of nitrate, the black bar, and each time we added phosphate, it decreased significantly, except when the addition of ammonium. Over here in ammonium concentrations, there was almost, not even detectable initial amounts. So the only time there's ammonium over here is when we added it. So basically, there was a boron limitation at Avon. because phosphorus and boron had the highest chlorophyll A concentrations, it supports our hypothesis that the lack of boron could be the reason for no heterosis. And studies have found that boron is needed for heterosis formation, so this could indicate that there wasn't enough boron there for them to develop heterosis. Phosphorus and iron was the second highest chlorophyll A at Avon, so it supports our hypothesis that iron is needed for the take-up of nitrate. If you look here, this shows nitrate again. When we gave them phosphorus and iron right here, the nitrate concentrations went down significantly. So studies have been done that show co-limitation between phosphorus, ammonium, and iron. And then phosphorus ammonium had the most chlorophyll A at phos, right here. And overall, phosphorus is the main limiting nutrient in all algae. If they don't have phosphorus, they can't grow. And nitrogen is often the second limiting nutrient. So studies have been done that show that phosphorus and nitrogen can co-limit. Phosphorus and boron was the second highest at SOF right here with chlorophyll A concentrations, which may indicate that boron may have also been limiting. And then the addition of phosphorus allowed the uptake of nitrate, which is shown over here. So in conclusion, trace metals affect heterosis formation and nitrogen uptake. So they are playing a role, but still algal growth is mostly phosphorus and nitrogen limited. So some important questions this could raise is trace metal clean sampling and laboratory methods and why those are important in the study of cyanobacteria. A lot of the sampling techniques have metals on the actual devices for sampling, and in the lab sometimes people clean bottles with metal bottle brushes, and those are all introducing new metals into the water that may not have been there in the first place, so that could skew your results. Another question a lot of people might ask would be, will the liquospermum develop the heterosis? So as they're living in the water, they're using up all the nitrogen that's there. So if they don't develop their heterosis, then they're going to die. So it'd probably be a good idea to do that. These are my acknowledgements. I'd like to thank all those people.
Is boron naturally occurring in the lake, or is there some sort of
It's naturally occurring, but this was a question I also had. I think I would need to do more research on where these trace metals are coming from, but I think people know where they're coming from, but it's just they're being introduced to the lake, and there's no way you can really limit it because also the concentrations of them are astronomically low compared to phosphorus and nitrogen. So okay.
Are there any other reasons that we might want to try to prevent heterosis formation? It seems like you found that you could limit heterosis formation by limiting boron.
Right. Yeah. Well, also, I guess it's almost impossible to limit because the concentrations are so low. People have said that, Dr. Kane said that there may be boron introduced in fireworks, and I don't know if we'll ever see fireworks go bye-bye, so I don't know if any limiting can be possible. It's just generally good to know, what they're using and what's important in their growth.
Why did you choose to take your water samples from two different sites that you did?
Well, initially we were supposed to do 2 experiments, both sampling at Avon, but the second time we went out, we got caught in a storm, and so we couldn't get to it. Well, yeah, 2 storms. 2 storms same day. So, and the first time we went out, we actually got caught in a storm, but we did make it to Avon. But yeah, so we could only get to as far out as Lorraine, And I actually collected all of my water from Lorraine, and then we got to Soft, and we thought there was more chlorophyll, or more algae there, so we had to dump out 35 liters of water, and then refill it up. It took a long time.
And I got 33 of those with buckets, and she got the last two, but I was not happy to see that poured all back in the lake.
Sorry.
No, that's okay, it's for science.
I've noticed that you never had a nice sunny picture.
I know, yeah. I didn't see that side of Lake Erie this summer.
What was in your all treatment? I forgot. Is that
It was a mix. Well, it was like 1 milliliter of phosphorus, 1 milliliter of ammonium, boron, molybdenum, and iron. So it got all of the nutrients together.
Did you have an idea of why you didn't see at least as much response in that treatment as you did for like the one with phosphorus plus boron?
Yeah, that's a very good question. It was also interesting that like the addition of ammonium seemed to decrease nitrate uptake. I don't know. I mean, I guess it's using ammonium over nitrate, but yeah, I don't know. It was kind of strange since we had such a low amount of algae in the water in the first place. It was very low concentrations because there's no blooms. There were no blooms out there. But it could be just a freak occurrence, or maybe it's replicable. I don't know.
So in a year when you had a bad toxic bloom, does this suggest ways you might go out and try to treat it?
Could. These are questions I have myself. I don't really quite understand where trace metals are coming from in the first place, which would be a good place to start looking. I don't know if you can go out and extract boron from the water, or, I don't know exactly, but it's definitely, my research supports all the previous research that has been done, that phosphorus is the main limiting nutrient, and so that's what the focus should be on, is to decrease phosphorus input. Yeah.
If you know that boron is a problem, then instead of trying to treat the boron, you might choose to treat whatever brings the boron in. It might be a protein-based approach to treat the algae.
Yeah, definitely. No more fireworks. No more fun. Yeah.
Would there be a way to interrupt the chlorophyll? So I know chlorophyll is not a very stable molecule. Is there a way to try and break it down so it can't be involved?
Are you talking about like going out in the lake and like dumping something in so it messes up the chlorophyll, or?
Just start out with an experiment.
I mean, yeah, but there's other plants in the lake that need chlorophyll, and there's other types of algae that use chlorophyll, like green algae, that are like plants and animals need. So I don't know if that would be a good idea.
Comment, but one exciting thing about finding these micronutrient limitations in the lake, especially the ones that affect the nitrogen cycle, or specifically nitrogen fixation, is that it goes back to the question that I asked Laura. Could we remove nitrogen from the water? And if these things are actually limited by boron, then in fact, if we remove nitrogen, we might actually be able to have a response, because they won't be able to get their own nitrogen from the nitrogen fixation process. So we might spend a lot of money and actually get a lot of benefit, and that's what we're trying to learn.
All right, our next speaker is Brenna Ortega from Cornell University. She's going to talk more about nitrogen, but it's going to add a little bit, just some more make it a little bit more complex for everybody. So Brenna's a junior at Cornell, and in her first sentence of her application for Stone Lab, she said, "I'm a native Clevelander, and go tribe." And then in the final line, the final paragraph of her application, she wrote, I rode in Cuyahoga River. I've been the burden of all burning river jokes, and I forget there was one other comment.
I've eaten the Lake Erie walleye.
Oh yeah, eat Lake Erie walleye. And but besides, with that part of the application. In the middle of the application, it was really good. She had a bunch of she had a bunch of other internships. She was out in New Mexico last summer. She had an internship at Ithaca College and at an environmental firm in Stowe, Ohio. So, very good.
Thanks for that introduction. So this summer I worked with Dr. Chapin on a project concerning eutrophication of Lake Erie. We looked at the effects of various nitrogen compounds on algae growth and toxin production. And what I didn't know when I was making this presentation was that everybody who came before me was going to tell you exactly what I'm going to tell you next. So in case you didn't get what eutrophication is the first 3 or 4 times, I'm going to tell you again. Eutrophication can generally be defined as excess algae growth in a marine or a freshwater ecosystem. Around here, it generally manifests as a bloom of cyanobacteria or blue-green algae, and generally looks like this at its worst. It creates this sort of green surface scum that isn't too pleasant to look at. The previous research has shown that these blooms are mostly limited by phosphorus and nitrogen, with phosphorus being the main limiting nutrient. Around Lake Erie, about 70 to 80% of the watershed is agricultural, so most of the phosphorus runoff comes as fertilizer after large rainstorms. Eutrophication doesn't just affect the Great Lakes, however, it's a global issue. There are various types of blooms that can manifest in Gulf of Mexico, off the east coast of China. This is a red tide off New Zealand. And these blooms can all produce negative effects on water quality. So they produce toxins. Cyanobacteria produce liver toxins and neurotoxins, and they can create dead zones. So what happens is a lot of people hear dead zones, and they think that it's a direct effect of eutrophication. But what really happens is that as the algae die, they're broken down by bacteria. And bacteria are like us. They breathe in, well, more or less, they breathe in oxygen, and then they exhale carbon dioxide. So they're taking out a lot of the oxygen that's in the water, depleting it from the hypolimnion, where there can't be any mixing to get new oxygen back into that area of the lake, as was mentioned earlier. So that can create fish kills. And the algae can also create poor conditions for aquatic life because it limits the amount of light that can get down into the lake and generally isn't very good food. There are also some human health concerns. Last year's bloom was not the largest bloom that we've seen, but it was actually pretty intense in that it caused Carroll Township to shut down its water treatment plant for 2 days. There's a certain level of toxin that we can ingest without getting sick, and Carroll Township exceeded that limit, and the city of Toledo came dangerously close to doing the same thing. As you can imagine, these blooms are also detrimental to fishing and tourism, because if there's no fish, then you can't catch them, you can't sell them. There goes that. And tourism in general, people come here for the alcohol, but they also come for the beautiful views of the lake, and nobody really wants to go to a beach where, you can't swim in the water and it's not very pretty to look at. Around the Great Lakes, we've seen a resurgence in eutrophication events in the past decade that we haven't really seen since the 1960s and 70s. Lake Erie is the shallowest and most biologically productive of the Great Lakes, so we've kind of borne the brunt of most of the eutrophication events. This is what the average, or the extent of the eutrophication events has been in the past about 12 years, with this year's forecasted bloom. So you can see that it's pretty similar to what we had in 2013, and about half of what we had in 2011. But there's still this big scary error bar that says that it could get pretty bad this year. So what we looked at was the role of nitrogen in toxin production of cyanobacteria. We wanted to test whether the different compounds of nitrogen commonly present in Lake Erie and the different dosages that they can arrive in affect overall algae growth, toxin production, because about 14% of microcystin, which is one of the toxins produced by cyanobacteria, 14% of that is nitrogen. And then we also looked at how these compounds affect the activation of toxin production genes. The dominant cyanobacteria in Sandusky Bay, which is where we did our sampling, is Plankton brixigardii. So that was what we looked at when we went out sampling. On July 2nd, we went out to Sandusky Bay and collected about 35 liters of water to take a look at how this plankton brix would respond to nutrient enrichment. We separated our water into 24 treatment bottles, 24 one-liter treatment bottles, some water left over, and added nutrients. So we had a control. We had three where we added phosphate, and then we did nitrate, ammonium, and urea in 2 different dosages. So we had a set that was a single high dose or a single high concentration. And that was meant to simulate an extreme loading event where a lot of phosphorus and nitrogen would come into the lake at one time, such as after a rainstorm. And then we also had a low-dose continuous treatment where we would add to it every 4 hours. And we put all of the bottles into an incubator for 48 hours and continued the experiment. So every 4 hours we would come back to the lab and add nutrients to our continuous treatment bottles. So we had to come in at 10:30 at night and 2:30 in the morning and 6:30 a.m. It was a blast. When we would do these nutrient additions, we would take all the bottles out of the incubator, regardless of whether we were adding to them or not, and shake them just to keep things consistent. At the end of the 48 hours, we did several tests. So we did some filtrations for dissolved nutrients. We looked at phosphate, nitrate, and ammonium using the nutrient autoanalyzer that was mentioned earlier. We also did a test for urea by a spectrophotometric method, and then we did filtration for chlorophyll using the DMSO method that has also been previously mentioned, so I don't really need to tell you what we did there. And we looked at microcystin, the toxin produced by cyanobacteria by the ELISA method, which produced some really lovely, pretty colors, and filtrations for RNA. What we looked at for the RNA was how quickly the cyanobacteria was able to turn on the toxin production genes. So we did filters at time 0, 1, 4, 8, and 48. That is going to be determined by an off-site lab, so we will get those results later. What we found was that nitrogen overall stimulated algae biomass growth, but there was no real pattern apparent among the different dosage concentrations or the different treatments. So, like Katie's experiment, we used the Tukey test to show us, the different letters represent different statistical differences. So we did have a statistically different results between urea and then the nitrate and ammonium. However, within the context of our experiment, there is a difference, but it's not meaningful applied to Lake Erie. Toxin production was also stimulated by nitrogen. All the compounds showed an increase over the control and phosphorus treatments, and ammonium overall had the lowest mean. There was a slight increase from each of the single high concentration dosages to their respective continuous concentration dosage, but it's not enough to be statistically significant, so we can't really say that a single high loading event versus the continuous entrance of these compounds into the lake really one over the other doesn't make a difference. We also looked at the amount of toxin produced per the amount of chlorophyll produced by every cell. And we didn't find that there was a difference among any of the treatments. So the reason that there are no letters at the top of any of these bars is because they would all be letter A. Microcystin, instead of being limited by a particular type of nitrogen, was instead limited just by overall biomass production. Our dissolved nutrient analysis showed that all of the nutrients were below the detection limits after the experiment had ended. So the cyanobacteria ended up consuming all the nutrients added, regardless of what we added and in what concentrations. Ooh, forgot to mention this, but at the end of the continuous low dose, all of the concentrations there were equal to the high concentrations at the end. So we remained nitrogen limited throughout the experiment for all the treatments. So what were our results? I'm sure you're all dying to know. we found a significant response to nitrogen enrichment overall. So that means that, nitrogen does play a role, especially concerning chlorophyll A and microcystin. So biomass production and toxin production both increased with the addition of nitrogen. But there was no definite compound that really increased either of them more than the others. There was a slight increase for the low-dose microcystin production over the single high dose, but again, not statistically significant. And the toxin production per cell was limited by biomass and not by particular compound of nitrogen. Also, all the dissolved nutrients were consumed by the end of the experiment. So what all does this mean? Well, in a global context, we're seeing a trend of increasing nitrogen use in fertilizers. So we can expect to see more toxic, more widespread blooms as this line keeps going up like that. And there it goes. We've also seen a switch from ammonium and nitrate-based fertilizers to urea-based fertilizers, mostly for economic reasons. But my results show that this is not going to affect the overall toxicity of the blooms. I really like the laser pointer. I can't help myself. The entrance of additional nitrogen, as we saw in the previous slide, will exacerbate blooms. And we were wondering, well, will minimizing nitrogen loading help? Possibly. As Laura mentioned earlier, if you remove nitrogen, then you're just going to end up with nitrogen-fixing cyanobacteria instead. So if we're going to decide to regulate nitrogen for whatever reason, we should take all forms of nitrogen into account because none of them really has an impact that's greater than the rest. I'm going to be staying here at Stone Lab for an additional 3 weeks to repeat our experiment, and we're going to be looking at Microcystis aeruginosa in Maumee Bay, which is the dominant cyanobacteria there. So it would be interesting to see whether the two different types respond differently to the same treatment. Because obviously, Planktothrix isn't the only cyanobacteria that's causing issues. There are many other different types. They could all have different responses to nutrient enrichment. We're also looking forward to getting back our results from the RNA analysis so that we might be able to tell from that if the toxin production genes depend on a particular dosage of nitrogen to be turned on. I would like to thank the Friends of Stone Lab and Ohio C. Grant for funding me and allowing me to be here this summer, Dr. Justin Chapin for guiding me in my research, and Brianna Zellner and Victoria Simons for helping me out in the lab, and Captains Craig and Art, again, for taking us out to our sampling site. What was that? Let's go. Dance, Bill. Do it. Do it. Does anybody have any questions? Yes, Dr. King.
So it seems to me, unless, and I might not have read the latest papers, that there is still a debate in the literature whether the form of nitrogen versus just absolute amount affects microcystin production. Do you have enough of a feel for that from doing this? I mean, yours says, no, it doesn't matter what type of nitrogen, but what have others said?
Well, I'm not really sure. I really only have my results to go on in a couple papers I read 3 or 4 weeks ago. But, I suppose it does depend on what the concentrations already are in the lake, and generally where these compounds are coming from, what other compounds might be limiting. So, to sum that up, I'm not sure.
Yep, 3 more weeks to find.
Yep.
Do you have a hypothesis about the RNA results?
Well, based on the results that I've seen so far, I don't think that we're going to see a difference between the compounds of nitrogen or the dosage amount. But when I first started, I thought that it was going to be turned on by a sudden high loading event.
So that's your hypothesis originally going in?
Yeah, but now having learned things over the course of 5 weeks, changed my mind and say that I don't think that's going to be true.
what do you feel about, I mean, it seems like you're still getting some indications that limiting nitrogen could control the toxicity of blooms. Somebody asks you that question, how do you respond?
Well, as far as Planktothrix is concerned, I don't think that limiting nitrogen will really affect the toxicity. It seems that there is some, nitrogen does enhance the amount of toxin produced over just having phosphorus. So I guess that there might be some co-limitation between phosphorus and nitrogen, but as we've seen in some of the previous presentations, just removing nitrogen isn't going to help because that will just favor nitrogen-fixing cyanobacteria. So instead of having a Planktothrix bloom, we could end up with an Anabina bloom, and limiting nitrogen then would not be helpful. We're working away on making sure they can't form heterocysts, and we'll collaborate. In 7 years, and we have PhDs.
Thank you very much. Let's take a break until 8 o'clock, and we'll come back in.
You got it.
We're going to get started with the next presentation. I'll turn it over to Justin.
All right, so our second set of presentations are going to focus on the vertebrates. So we're done with green goop and oxygen and nitrogen. We're moving on to fish and snakes and birds. So I'll let. Okay, I guess Abby.
Yes, there was a slight change of plan.
Okay.
My name is Jeff Steinhardt. I was Abby's mentor for this project, and my wife and I need to leave, so that's why Abby's going to go first now. Abby's a senior majoring in fisheries and wildlife, with a concentration in fisheries at Lake Superior State University up in the UP, Michigan. That's how I know Abby, and so when I found out she was coming to Stone Lab and wanted to do an REU project, we had already talked about some research ideas when I was in Michigan, so anyway, I had to fight off some ODNR people to work with her, but everything worked out great, and she's going to talk to you today about colonial nesting sunfish.
Okay. So ever since I was a little girl, I've been into fish, and this fish in particular, I used to fish with my grandpa and my dad, so this is kind of something that I wanted to develop younger, but as a college student too, so it's nice that I finally get to study them and not just fish for them.
You may have to use the arrows on the computer.
You have to turn the clicker on. It was actually just my delay. My mom's coming. Oh, she's right here. So perfect timing. So there are two types of nesters, and then on the left is a solitary nester, which is more common, such as smallmouth bass. But then on the right, there is colonial spawners, and that is what bluegill are. So the difference between the two is that the solitary nester will only have itself to guard off all the predators, while the bluegill spawning habit, they can all attack each one of these fish and kind of move with predators to overlap protection. So it's said that being in these groups, it's energetically costly, but it helps predation risk. So parental care. So for the male, the role is to clean the nest, provide the eggs, and protection. So why is it beneficial? There's safety in numbers. Like I said before, they can attack as a group to predators, and the females are attracted to larger colonies. I was joking around, wouldn't it be cool to be a fish? Because all these men line up, fill these nests, multiple ones, so that the women can be picky, lay their eggs, and then the males do all the work.
That's definitely not how it is in the real world. Sometimes I wish I was there. So the males provide sole protection of the larval fish. The cost, however, is associated directly with density. Since there are so many fish spawning in one area, they attract predators because they're moving around and they actually release an odor. And then the chances of diseases increase because they are so close together. So Dr. Steinhardt, my advisor, has also worked with a past REU student. Her name was Leah. She recorded kind of the same thing. She wanted to look at sunfish, so the difference between bluegill and rock bass. And there was reason to believe that there was intracolonial and interspecific competition, which those terms mean intracolonial, meaning the male bluegills will eat other male bluegill eggs. And then the intraspecific means of the same species, so bluegill eating bluegill eggs. So we thought of this great idea to study the colony in general and determine who are the nest predators, what is the difference between the colony size, and so we did that by measuring small to large and measuring nearest neighbors. So our study site was directly right across the lake at Wall Harbor. I didn't know what the name of it was called until I put up a map. But it's actually right here where there's minimal boat traffic, and that is actually the exact place that Leah did her study in 2009. When I was getting attacked by a fish, the fish actually moved its tail real fast, and I found one of her old rocks. So it's kind of cool. So the survey design, we use an underwater camera to record predation, and that is what this is in the water right here. So when I set the camera out, I set it out, came back onto shore, and then waited 5 minutes to not only let the water settle down, but to let them get into their own natural behaviors, so no human interaction. And then I caught the male off, and sometimes I was successful, but sometimes I wasn't. And then we placed the male in the bucket, and then that 5 to 10 minutes, depending on, we had some problems with the camera when it ran out of battery or film. So those are all things that you're going to run into. And then, so like I said before, we separated colonies. The separate colonies is defined as 5 meters away, so we actually had 5. Large colonies are greater than 5 nests, smaller, less than 5 nests, but we scaled it down to less than 3, because that's what we saw. And then we measured the distance from nearest 2 to 5 nests. And then, so after we had all these days of data, I actually had to sit in a room. It's actually one of these. The bird guy's room. Yeah. I actually had to count and timestamp when predators entered the nest. And so I have an example, and I think it's better to show you than to explain it. So in the beginning, I'm going to catch the fish off of its nest, and you'll see what happens after. That was me catching it. And then as time goes by, you'll see all the predators. So those are, we classify those as other bluegill because they're not parental bluegill. And you can see they're kind of chasing all the predators off so that they can have the nest for themselves and to consume the eggs. So it's kind of fun to watch.
I really broke it.
The little green globe down there.
It's green globe, okay. I think it broke it. I didn't have to turn that on or off. So our data collection, we had 5 colonies in total. 3 were large, these 2 right here, and 2 small, this one and this one. And then there was 20 nests total that we were able to sample pre-catch and after catch. And there were 17 bluegill and 3 rock bass. But since the rock bass were ultimately providing protection and receiving protection from the bluegill, we included them into our study. So of the 136 species, there was those species, but they entered 136 times. There was parental bluegill, other bluegill, goby, and other, and we clumped rock bass, smallmouth bass, and yellow perch into the other. And our results were pretty cool. So this axis is minutes after the male is removed, and then the number of entrances. And so you can see in the first 3 to 4 minutes that gobies have entered the nest. But then, as you saw in the video, the other bluegill and parental bluegill in blue chased them off and actually started consuming the eggs for themselves. And then here's just another diagram to show that the blue is the predators that entered the nest were actually the bluegill, which were the most active. And then as time goes on, as I stated before, that the bluegills will actually enter the nest, chasing off the gobies, so the gobies didn't increase as time went on. And then we had this relationship between large colonies and small colonies. And then the nearest averaged 3 neighbors, but they were significantly correlated, so they were kind of lumped together as the same in statistical data. So in this one, large colony, you can see that this is where most of predation comes in. And then here's a small colony, and you can definitely tell the difference. This one has more color on it, meaning that there was a lot more different species as nest predators than the small colony. And then so to conclude our analysis, we did an ANOVA comparing bluegill predation to the average nearest 3 nests, and that was not significant. But the minutes after the guarding male was caught was significant. So minutes afterwards, they fought off the gobies, and then they were more prevalent in the end. So, and then the relationship between minutes after male capture and average nearest nest was significant because they are going to swim to their nest to the others in order to consume the egg. So before I get to the acknowledgments, the next studies that we need to look at is, for me in particular, I need to combine both my data and LAIS data to see if there's an overall trend and compare that to goby population data to see if it was just a year, if there is actually, if goby numbers are decreasing. And so that could be another study in itself, is just the goby population. DNR does trawls, but they aren't accurate in regards of gobies inshore. So my acknowledgments, I'd like to thank Dr. Steinhardt and Dr. Aaron, both because they were my mentor and my advisor at Lake State. And they're a package deal, so if one's sick, then the other one has to stay home with the kids. So they're both equally important. Dr. Simon reviewed my papers last night, which I am grateful for. Stone Lab staff, in particular the groups that we've had Thursday nights to help with talks. Friends of Stone Lab for making this possible for me to be here. The Ohio State University, I got corrected, it's the, and Ohio Sea Grant. Questions? Yeah, go ahead.
It almost seems, from looking just, cursory look at this, that there's an advantage to the bluegills that nest, in small, or almost solitary, or?
Exactly. I need more data. That's why I want to combine Leia's data. But it seems to be almost beneficial for them to be solitary nesters or to make that transition. But I think it all has to do with DNA, because it has been said, and more needs to be looked at this, that the neighboring bluegill males are genetically related. So maybe they think that it's okay to eat the other offspring's eggs because they are related somehow, so it'll give them energy to take care of their eggs, which are maybe 1/8, 1/16 theirs. So I think there's a whole other relationship and fundamentals of nesting and colony sizes that need to be determined. So I'm going to enter more data from Leia's and to see overall if it is beneficial.
Would you, based on what you've seen, it would seem that overall mortality rates would be much higher in the root nesting area?
Yeah.
Okay.
From their own kind.
Yeah, well, and, but even, from their own kind, but from any kind, actually.
Exactly.
And it's all, I'm just wondering if there's bluegills are really known for stunting and overpopulating and all that kind of stuff, and this is almost some kind of a natural selection method here to, as the population gets bigger, the nest gets closer together, you get all kinds of increased mortality.
It's interesting. I'm going to compile more data and make more graphs to see how in the years it's changed, because it does have to do with also the invasive brown goby preying on their eggs. I watched a couple videos from Leia's because our tape didn't run through, but I was like, oh my gosh, there's so many gobies. Like, I kept counting them and counting them and counting them. In my study, only 4 out of the 20 nests had gobies in them, which is significant.
Would there be a certain change in context that would maybe cause a shift in what motivates them to colonize, like, as opposed to, like, because right now they're colonizing out of selfishness, but would there be a certain change in the environment that would drive a change to colonizing out of altruism? Some other species colonize out of altruism, others colonize out of selfishness.
They are, bluegill are socialized nesters. I think they like the interaction, but I do think it'll have to do with DNA analysis, because if they are social, it would make sense to bond, with your cousins, your brothers, and just to keep the DNA pulled together.
Do you think there's a relationship between the size of the bluegill and maybe the ability to be more solitary in your nesting behavior, or do you think it's really more of a relationship among just the habitat that's available out there?
I don't think it's habitat, but I do think something that has to do with it is the size of the eggs. I read a paper that the size of the eggs, as they increase, the more parental energy they expend. I think it has to do with their eggs. Also, I don't know if they can sense storms or whatnot, but if it's not going to be good habitat for the eggs, they might not put so much energy into it.
When you pulled the bluegills off the nest, what did you do with the male?
We put them in a bucket full of lake water.
And then you put them back eventually?
Yeah.
All right. And they were off for 10 minutes?
5 to 10.
5 to 10. Did you measure them or anything like that?
No, we didn't.
Okay.
If I could go back in time, I would like to measure them and maybe see the correlation between their particular size, their nest size, and their nearest neighbor distance.
Well, or even what Dr. Simon was thinking, did it look like the ones that were in the solitary nest tended to be larger males than the ones that were in the group nests, or?
Actually, the video that you saw was number 30. It was the biggest, probably bluegill I've ever caught. It was bigger than my hand, probably. That's no fish story. But it had the biggest nest, and it was in the largest colony.
Oh, okay.
announce another Michigander. And I didn't know Scott was a Michigander. It wasn't the reason I selected him, but it might have been. Scott's from Albion University. He's, one thing about Michiganders, they always have to show you where they're from with their hand. I won't do that. I'm not going to do that. So Scott's from about the middle of the palm, and he grew up actually very close to where I grew up. So we have a lot in common. And we share a love of the same kind of music. So with that, Scott will be presenting his research. He's a senior. He'll be staying on for the next couple of weeks and continuing. But he'll be presenting his work looking at aging growth of brown gobies.
All right, thank you, Dr. Simon. So I'm pretty much here to tell you guys what's the big deal about this little fish. It's only about 4 inches long, but it can have a huge economic and ecological impact. Basically, what we're seeing, we need to understand the life history of this fish, because it plays a huge role not only in our native, or its native region, but in our Lake Erie ecosystem as well. If I could just ask all of you guys, I want to pose a little bit of a challenge to you, just so that you realize how prevalent this fish is. If you want to go and strap on some goggles and hop in the water, I can guarantee this will be the most prevalent fish you're going to see around here. They're everywhere, and I know it's touched pretty much everyone's project. Abby did a great job mentioning the predation, how that is a major role that the goby plays. So I'm going to move into a little bit of the background, and I have to admit I'm a bit of a history guy, so if I can get this going, I'll hopefully be able to tell you a little bit about the history. I guess I can start off on that. Alright, so the brown goby is a euryhaline fish species, which means that it can survive in both saltwater and freshwater ecosystems, which gives it a huge advantage when it's moving into new regions. Thank you. And it's native to Central Eurasia, primarily the Black and Caspian Seas, highlighted in green over there. But it's managed to move its way around to all these different regions in Europe as well as the Great Lakes. And you're probably thinking, how did that little fish swim all the way across the Atlantic? It didn't. It took a cruise. So basically, the larvae of this fish are pelagic, which means that they are primarily at the surface of the water, and they get sucked in with the ballast water in a lot of ships. And this ballast water is emptied out when they enter into the Great Lakes and other regions when they need to go into shallower waters, and they're essentially going and releasing and dispersing these in different locations all around the world. And we're first seeing gobies in Lake Erie in 1993, with the first being noticed in 1990 in Lake St. Clair, just above Lake Erie. And they're currently still spreading throughout the world. In 2011, they noticed the first ones in the Rhine River in France, as well as they're moving into the Finger Lakes region of New York. And the reason I guess they're doing so well is because they're both euryhaline and they're very tolerant to these degrading environmental conditions. And I know that the first 5 talks you've all heard plenty on how Lake Erie's pollution and other environmental impacts might influence the goby to be so successful here. So the ecological significance of this fish is not only just, I know when a lot of you hear invasive, you think just horrible, awful, worst things it can possibly be, but it has actually had some positive benefits in our Lake Erie ecosystem. It's a plentiful food for game fish. They're not very fast. They can just get picked off by walleye and bass and provide excellent food for them, as well as the Lake Erie water snake, where it composes about 90% of its diet now, and has actually helped it recover from being endangered. And it also feeds on coiga and zebra mussels, so it's helping keep those populations in check as well. But it does also have some negative consequences. It has outcompeted the mottled sculpin pictured above and log perch below, and it feeds on native fish eggs, like Abby had mentioned, which includes game fish, so it could have a huge economical impact on Great Lakes as well. And it essentially dominates benthic habitats. It's very aggressive and can outcompete many of these other species. So the objectives of my research, we just want to come to a better understanding of this fish and what sort of management plans we can possibly make from that. It's here to stay. It's been here for 20 years now, and it's extremely prevalent. So you can't just get rid of it. You need to figure out how to manage it. We're trying to figure out how the growth rates vary year by each year and by the age class, as well as the overall condition of the brown goby population, meaning the health of the population, essentially. And we're looking at samples going all the way back to 2006, so we're hoping to see changes in the brown goby population structure over the years as well. So in order to do this, we had to catch some fish. We primarily used seine nets, pictured below, and outer trawl above. Outer trawl is used in deeper areas, just to kind of pull out all the fish from the benthic areas around there, and then the seine nets are used inshore. We have samples from 2006, 2008, and 2014, as well as all the years in between that almost, that we have not really gotten to checking out yet. And we've gone over 459 individuals, and I know these all look like fantastic, just outdoors, doing all that sort of stuff, but the majority of our work was in a lab, blasting AC/DC, just going around, and we were taking the total length measurements, standard length, wet weights for all of these ones, and taking scale samples as well. So a lot of hard lab work, but we managed to get through that. So with that data, we were able to find some really interesting things. We plotted the wet weight against the total length to try to figure out the condition of the species. In 2014, we did this for each of the years. I'll go into that later. But as you can see, our trend line doesn't really fit too well, and as much as I really don't like throwing extra math into things, it's really simple. We just put it on a log scale. We have a much better fitting line now. And with that B value that you see in red over there, 2.765, that's actually from Fulton's condition index. And what you would expect to see in a population that has just an average stable condition would be a value of 3. Fish above that are essentially putting on more weight than they are length, meaning that in fish terms it's healthy. Kind of the opposite for humans sometimes, but for fish that's how it works. And with these values below 3, that means they're putting on more length and kind of growing lean in length. They're not really putting on the weight they should. And that's what we're seeing in 2014. So we wanted to check up on the sex distribution throughout all of the years and see if maybe the ratios had changed. And our overall ratio was actually statistically significant, but when we broke that down further, we found out that it had been heavily skewed by the 2008 data, where there was almost a 1-to-2 ratio of males to females, which is a very interesting population structure, especially compared to the other years where they're just about equal, not statistically significant. So we broke down the length weight distribution into males and females, both providing very similar condition index values. And then we decided to go and compare that to our other years. 2008 is pretty much significantly lower to that. It's not drastically lower, but it is indeed lower, which means that the condition was actually worse back in 2008. But the real surprise, they're fairly similar. The real surprise comes when we check out 2006, where they actually had positive allometric growth, meaning putting on more weight than their length. And threw off the scale, I guess, a little bit on here, but you probably get the picture. 3.32 was our value for that, meaning that the population was actually had a much better condition back then. So the other part of our experiment, we were using a scale aging technique dating all the way back to 1898. It's a very old technique, and it's been proven to work well if you take the right precautions. So what we've been doing with our scale samples, we mounted those on slides and then took a look at them under a microscope using an ocular micrometer, which are the measurements that you see there on the line. And we measured the total scale length, measurement from the focus to the edge, and the focus, if I can find this laser, is right over here, and that's essentially the butt of the scale when it first starts forming. Now, gobies and other fish are a lot like trees in this respect that basically what you're going to see is each of these lines represents growth in the scale. And during the winters, you would see these lines come together closer because there's less resources, less growth. So you're going to see kind of darker trends in the line. And sometimes they're difficult to see, but you get an eye for them after a while. So we're having one right over here, and then another one over here. And each one of those would represent a winter that this had survived. So just to kind of highlight them a little bit more for you guys. And those are called annuli, and those just form yearly at the winters, and that's what we measure out to figure out how old the fish was at a certain age. So by taking the ratio of scale length to the total length at capture and comparing that to the annulus length, we're able to go and back calculate the lengths at different ages for each of these fish. So this is our age and growth data for 2014. Basically what this will mean is that at age 1, even though these fish are age 3, we back calculated them to being about 27.25 millimeters in length. And their capture length is about 74. So what you'll be noticing here is that these numbers are relatively similar for 2014. And we're actually able to go and use this. I guess I'd do that in there too. Graphs are a little messed up here, but bear with me. We'd have the number of individuals here. And what we would be seeing here, we were trying to validate these lengths, validate these aging techniques using length and how frequently we were able to capture fish of these different total lengths. And just to prove that we're not going completely insane when we're seeing these little lines that are barely there, we took the maximum and minimum ranges for all of these year classes, and we've highlighted them over in the red, and they overlap with all of these trends that you're seeing in the graph, all these lines, essentially the curves that you're seeing, the peaks. And you'd want to see the middle of the peak, essentially right in the middle of where you would predict that age to be. And we were fairly accurate with all of this, so our scale aging technique did work. This would be age 1, age 2, and age 3. And if you notice, age 3 is longer than age 1 and 2, but that also is given that they could have been different amounts of growth in age 1 and 2, so you're going to see a wider range of fish at age 3. So for our age and growth comparison, we compared these to our 2008. We compared 2014 to 2008, and what we're noticing is that a lot of these fish were fairly similar in their back calculated lengths at all of these. But when we compare it to 2006, not only are we noticing that the numbers in the back calculated lengths are greater for 2006, but we also have years 4 and 5, meaning that we found fish that were 4 and 5 years old in our 2006 sample jars, which is pretty interesting, meaning that we're not really seeing the age distribution as we had previously. So we've taken that data and calculated instantaneous growth for the population and mean individual growth. And basically, the higher that these numbers are, the faster the fish is growing. And although you might think that the 2008 ones seem really high and they should be the biggest fish, they're just growing really fast, but they're not growing for very long. They're not maintaining the same age structure as they had previously, and they're essentially living fast and dying young. So for the mean individual growth, we've essentially just taken the population growth reflects all members of the population, whereas the mean individual growth is just your average, and they're fairly similar. So I won't go into too much more on that. But when we compare it to 2006, we're seeing that the growth rates actually have been slowing down, or have previously been faster at a point, but they're not maintaining the same sort of speed to them. They barely overlap, in fact, because we only had samples. We had one sample that was 2 years old and some 3, 4, and 5-year-olds. But if you look at the values for 3, they were indeed higher for 2006. So we were actually seeing faster growth back then, but it would always slow down as the fish grows older, as you can see. So we took the individual condition for all of these fish. This acts as the Fulton Condition Index that you saw before, where I was explaining with the 3 being isometric, above that being positive allometric, and below that being negative allometric. And although these lines don't fit very well, what you're seeing is a trend. These fish are essentially growing faster, and they're increasing their condition as they grow older, but they're not surviving longer. Whereas the fish from 2006 in blue maintain a fairly steady condition above 3. So that would actually be a much more stable population. And these guys are essentially just kind of burning out at that point and not really continuing as far as the age distribution goes. So this is kind of the takeaway message of this whole presentation. This is my key graph. What we're seeing here is we just plot the number of individuals by the total length. And although you might see these huge peaks, these are just done by the individuals as per custom, not by the percent. So what you're seeing here is the 2006 distribution is all across the board. You're seeing individuals as large as 140 millimeters and as small as 60, whereas the ranges for 2008 in green and for 2014 in red are much smaller, meaning that they're not gaining the same size that they once used to. So if you're like me, you're probably wondering, what's the big deal? Why is that? Like, how is this happening? What's going on with the population? And there's several theories that could explain this. Early maturation in these freshwater samples and introduced populations of gobies have been noticed. This particular study was done in Lake Erie, and it showed that they sexually mature our gobies a full year ahead of the ones in the native range. So they might be placing more of their resources into reproductive growth. And another study, Grula in 2012, showed that freshwater environments are only, the gobies in freshwater environments are only growing to smaller sizes, about 150, 160 millimeters, whereas the ones in saltwater environments, even in introduced areas like Danks in the Baltic Sea, are growing to 250 milliliters like they do, or millimeters, sorry, in their native range. Another theory would be niche fulfillment of the outcompeted species, meaning that the goby is essentially transforming its life history to fit that of the log perch and mottled sculpin that it replaced, including having the same diet and taking over the same habitat. It's also possible that the Lake Erie water snake, by selectively predating on larger gobies, would be kind of pushing these populations to smaller ones, as the smaller ones are much more able to hide in these smaller rock crevices and escape easily, and they don't quite provide as much bang for your buck, I guess, as the larger ones do. But the top 3 theories, those would have been things that we would have all seen originally when they were introduced. This is 20 years later. So those factors may have applied early on, but shouldn't explain the change between 2006 and 2008 and 14. So we're predicting that it's actually density-dependent factors having to do with the population growing and booming and then crashing, and that's what we've observed. Now, unfortunately, we've not gotten a chance to take a look at our 2007 samples, and that's going to be our key hinge point. We're going to find out if the condition was actually still going up at that point, or if that's when it crashed. And that should be able to tell us how the condition of the goby has been affected by its own population density, and hopefully we'll find out what sort of factors have been really affecting the population at this point, but it's going to require a lot more time in the lab and a lot more people to go and take a look at this. But brown goby is essentially transforming Lake Erie whether we like it or not, and we need to figure it out before it's too late. So I wanted to go and acknowledge Ohio Sea Grant and Friends of Stone Lab for funding all of this and making all this great things possible. I wanted to thank Dr. Simon. When I came into this, I pretty much knew that yellow perch tasted delicious, and that was my fish knowledge. now I've learned that there's this horrible thing called white perch, and you can't tell the difference between it and white bass, and don't bother with that. Wanted to thank Dr. Stanford for, I've never been so grateful in my life to get boxes full of dead fish, but thank you so much. We appreciate all the samples. I want to thank Dr. Reuter for helping us out with our presentations and giving us helpful feedback when we're doing our elevator talks. I feel like I'm much more able to explain this now. Captain Zard and Craig for taking us out and for not leaving us on Starve Island when we got there. I want to thank Tiffany and Tessa for keeping me company in this whole thing and just helping me out in every way possible in making this. All right, thanks everybody. Might have some time for some questions.
So, do you think that, like, slowly but surely hypoxia could be
It's definitely possible. I would say that it's far more prevalent to find the smaller, younger gobies in the inshore areas, the coastal areas. And what we're seeing, I believe Dr. Simon back in 2012 had observed that there were far less of these larger gobies being found in the deeper regions of the lake. So those larger ones might not actually be able to survive the hypoxic conditions, and that might be where we're seeing the major decline in the larger ones, just an ontogenetic shift, I guess, in where they choose their habitat, and they're just not getting lucky with hypoxia. Yeah, time for one more.
All right. If you have a hypothesis that you think this is being sort of density driven right now, which is a good hypothesis, certainly worth looking at more, would you expect that, if that's your hypothesis, would you expect to see this go up and down as the density goes up and down?
I would think so. I mean, populations just naturally are going to fluctuate, I would think, especially with changes in lake conditions. Right now, I think the goby is still trying to just kind of stabilize itself as a population. It's essentially boomed its way all the way through Lake Erie, and it's, I think, really going through a naturalization process where it's no longer really being the invasive attack that it once been, but where it's actually starting to kind of find its place in the ecosystem.
Very good. Thanks. Before you leave, make sure it clicks. Okay.
Quick clicks.
Well.
Thought it was easy.
Make this play for all. Go back to your era.
Did you select all slides and then hit that rotate?
I don't know how to select all slides.
Always right.
$30?
Yeah. I did not anticipate when I counted the slide number that it was gonna be stupid.
Jeff, what do you want to do? Do you want to... Kevin, let's just take a little bit of a time and go through and straighten them all out.
Go to show thumbnail. Hit
So then, do we have other time for questions for Scott then?
hit control.
Yes. So Scott, so where did all the gobies come from? Were they all just from around the island region, or do you have some from other locations throughout the world?
I'm actually not too sure about our 2006 and 2008 ones, but they're primarily from the Western Basin. I think all of them are from the Western Basin, in fact, and most of them are within the vicinity of the Bass Islands.
They correlate with our study, our water.
So they are pretty all, okay, that's.
Yeah.
Because that would help you with that fifth hypothesis, which is the water standpoint, because that might determine that around here, but if you sampled other places where that predator wasn't there, that could possibly be in it. And another one's more a comment than a question. When you're staying here for 3 weeks and working on this a little bit more, look into behavioral syndrome. And basically, in a nutshell, what that is if
if you have a behavior that's like aggressive, so maybe those individuals get more nest sites because they're aggressive, but also that aggressive behavior translates into other things like foraging. So maybe you're having selection pressure on those aggressive fish. So those ones that are aggressive so they nest better, but they're aggressive so they forage faster and grow faster. And maybe those are the ones that are being picked off by predators, and so you're actually selecting for the other behavioral symptoms. So take a look at some behavioral symptoms. There's questions back here, look at this.
Like, on a population scale, what's the difference between males and females? I mean, are males typically larger, females typically larger? Is there any difference in, I forget what basically like how bulky they are?
Yeah, so conditions, basically what we see for all of them is they're relatively the same as far as condition goes.
Now, why would the PDF this time automatically or anything like that? Then the previous time when I opened it.
I don't know. But, for most of our database, it's been pretty much stable. That's why I don't
Oh,
show
what's
the
the most
overall
retarded thing we can do it?
working figure images for most of our
Couple things I was gonna say at the very end of this too, and so this is a good spot. Dr. Simon, you've probably been the most successful in working with your REUs and former REUs to get publications from this. How many publications? Do you have any idea?
I think we're up to 6 now.
6 different publications have come from the REUs. We've probably had a dozen or maybe a little bit more than that students that have gone on and presented at the scientific meetings. That was one of the things that was already discussed. Really encourage you to do that. Along with both of those things, there are going to be costs. To make a presentation at a scientific meeting, you're probably going to have some travel costs. You're going to have some registration fees to get at that meeting. To make or to get something published, you'll have page charges. When you have those things, and for the advisors here also, through the Friends of Stone Lab, we'll cover those costs. We want you to go and make these presentations. We want you to work on the publications, and we'll cover those costs for you. And based on what I've seen, and really what I heard at the preliminary days when we were going through and first working on this, you should all be thinking about, at a minimum, making scientific presentations, getting those presentations on your record, and quite frankly, there's some really good publication possibilities that we've seen here. So work with your advisors on getting this stuff published. It's very much to your benefit as you try to go on and get into grad school. If you've already got a publication or two and experience making presentations, you're well on your way to really an outstanding scientific career. Are we set? Good.
Yep.
And I'm going to just briefly introduce Sarah. Sarah's advisor is Chris Vandergood, who's with the DNR, Division of Wildlife, based out of Sandusky. And he couldn't be here tonight for the talk, but Sarah comes to us actually she's from Florida via University of Montana and Missoula. So that's where she's taking her courses. So the furthest travel for our REUs this summer. She's got that plaque.
This is pretty much a pit stop on my way for eating my way across the USA. So, as you said, I'm Sarah Zundell, and I was working with Dr. Vandergood on analyzing post-surgery survival in Lake Erie walleye to see if technology will be more forgiving to me at this point in time. Well, Lake Erie of the Great Lakes is the most biologically productive and has a huge fishery that contributes greatly to Ohio's economy. And a good portion of this fishery, the money that's brought in from it, is from recreational fishermen. And obviously, you know, as much as I'm sure you guys like to eat the sheephead and everything here, these recreational fishermen aren't exactly coming up for that. This is what they love. So these guys bring in the money for the state. They're pretty much swimming dollar signs with fins. But in order to keep, you know, the money coming in, you make sure there's little baby dollar signs with fins. And the best way to assure that is to make sure that there is successful walleye sex. Unfortunately, this thing here has limited
In the Sandusky River stock, which goes up the river to spawn, the Ballvale Dam has cut off a large supply of potential spawning habitats, and they think they could increase productivity by removing the dam, which is in the approval phase now. It's been in the approval phase for a while. Apparently that's a pretty long process. But the overall goal is to describe the survival, spatial ecology, and spawning migration movements of walleye after restoring the connectivity in the Sandusky River. And my specific part of the project was to use acoustic telemetry data to determine the post-tagging mortality of 100 walleye collected from the Sandusky River stock. And this is what we like right here. This is more walleye on the way. The fish were collected with electrofishing gear on the 2nd, 10th, and 16th of April, and they were then taken to a set location where they, I'm going to murder the pronunciation of this, anesthetized. I'm sorry to all the fisheries people who know the correct pronunciation.
That was good.
That was proper.
With electrosedation, and then the transmitters were implanted in the fish. The fish were then held in recovery tanks until they were ready to be released. You know, if a fish is not recovered properly, you might let go of it, and it might wedge in between a rock, and there goes your fish. This is a picture where they took the fish to implant the transmitters. This is your final product right here. Get to what that is later. So we had 4 acoustic receivers located around the tagging site in the Sandusky River, and 2 receivers that were also located within Sandusky Bay. And I determined the survival by looking at how walleye moved between these receivers in the Sandusky River and Sandusky Bay. And these are the ones in the Sandusky River. This was the approximate release site, and fish were captured with electrofishing around here. So it was all kept very local, so you didn't have any, distribution bias. These are obviously the receivers in Sandusky Bay. And that is a receiver. Now, the part of it that's cut off there apparently is this was the process that I used to determine whether a fish survived or not. First, we determined which fish were harvested within the river. If they were reported harvested, and there was a $100 reward on the tag, and you could see by the, the purple external or the orange external tag that fishermen could tell. So they bring the transmitter in, return it, and we can tell which fish were harvested. So obviously, yes, they were removed from study to prevent bias. I assume that all fish were equally susceptible to catching. Was the fish harvested in Lake Erie? If it was harvested, we classified the fish as having survived because while it was in the study area, it did survive. So that was more of a validation. Then for the third qualification, was the fish detected upstream but never downstream? If it was classified as deceased. If it was not, then you move on to qualification number 4. Did the fish's downstream movement indicate mortality? If it did, and we'll get to that in a bit, it was classified as deceased. No, it survived. And then for a couple fish, they were kind of in the unsure category, needed further validation. So that was my final step, was the fish confirmed to be alive upstream? If it was not, if it showed patterns that were consistent with mortality, it was classified as deceased. If it was moving around, from a downstream receiver to an upstream receiver to a downstream receiver to an upstream receiver, it was classified as having survived. Now, 2 of these fish were classified as deceased based on the downstream receivers, which are what you're seeing here. Bet you can guess which 2 they are. Dead. And
why do you say that, Ms. Dutton?
Because they remained on the same receiver for an extended period of time. This is the Sandusky Bay is kind of a transition point. So when you see these guys that stay here for an inordinate amount of time, this one looks like it's bobbing away. You know, this guy looks, he just sank. So we came to that conclusion.
What time period are we looking at here?
This was between April and May. So my assumptions regarding survival were that walleye did not leave the system without our knowledge. You know, no birds or anything took our fish And then our second assumption was that harvested walleye were reported. There was a $100 reward, so there was motivation for people not to, you know, take the transmitter and throw it in the bush somewhere. Third assumption was that all walleye were equally susceptible to harvesting in the Sandusky River. And the fourth assumption was that fish did not die from tagging-related mortality after leaving the system, and violations of these assumptions would result in some bias. So 5% of the tagged individuals were harvested within the Sandusky River in our study area. And this is again with the assumption that these guys are all equally susceptible to harvesting. And then because of that, they were taken out of the rest of the statistical analyses to prevent bias. And afterward, 95 walleye remained. The adjusted total number of male walleye was 46, and the adjusted total number of female walleye was 49. So in typical fishery studies, when you're looking for total mortality, it's a sum of fishing mortality and natural mortality. What we were looking at is total mortality is fishing and natural, but also having the additional mortality related to the tagging process. And at this point in the study, we were counting all observed mortality in the tagging mortality process. The tagging mortality and the natural mortality would be separated later when these fish return up the Sandusky River to spawn. And then there will be no interference with them, so you can just see what proportion of them die in a completely natural run. And once the fish harvested in the Sandusky River were accounted for and removed, 23% of the remaining walleye, or 22 individuals, were classified as deceased. This is just showing overall trends. That's you have females having the largest proportion of survival compared to the males. It was actually interesting. I had initially predicted that the females would get hit harder, but in proportion to their populations, the males had a higher mortality. But I wanted to compare, to make sure this was statistically significant. So my null hypothesis was that female mortality rates equaled male mortality rates, and that both had a mortality rate not significantly different from the average mortality between the two, which was 23.65%. My alternate hypothesis, sorry, my voice cracked there for a sec, was female mortality rates are not equal to male mortality rates. I did this through a chi-squared test. Interestingly enough, it was not significant. The mortality rates for males and females was not significant at alpha value of 0.05. The test value came out to 4.44, where the critical value at that significance would be 7.815. And I concluded in this case that the difference in mortality may be due to chance, or we may not have a large enough population of walleye to see a significant difference. If it's a slighter difference, it's going to take a larger population before you observe it. And the patterns in observed mortality for length were the average length of the male walleye, just all of the ones excluding the ones in fisheries, harvested and fishing, was 514 millimeters, while the average length of the deceased male walleye was 501 millimeters, the smaller males. And there was functionally no real difference in females. The average length of the female walleye overall was 614 millimeters, while you saw a slight increase in the average length of the deceased female walleye, 618 millimeters.
And also, neither of these were statistically significant, difference in length between the deceased and surviving males and females. Interestingly enough, while neither was considered significant, the males had a much higher, relative significance than the females did. The females were much closer. It didn't seem like there was any determination of all in length relation to mortality. And this is, for this I divided up, we had, as we covered before, we had 3 different tagging and release dates. So this breaks down the mortality between the different release dates, April 2nd, April 10th, and April 16th. Not quite sure what happened in April 16th here. We looked up different, water temperatures, but it seemed rather random. The temperature on this day was actually closer to temperature, on this, and actually the outlier for temperature was on April 10th. So it didn't seem to make any sense as far as that. So one possibility is that it was later in the season, so these fish were more exhausted. They're up there spawning. It's a lot of effort for fish. The highest mortality rate, again, for males and females was observed on that last tagging day, April 16th. And males had equal mortality rates on both April 2nd and April 10th. The lowest mortality rate for females was on April 10th. Using chi-square test, I compared the average mortality per day to the mortalities for the average mortality per day to the mortalities for each of the days separately. And again, we didn't see any real significance there either. So this is, I wish it would show. So the natural mortality minus the observed post-tagging mortality. And again, that will be determined next season when the walleye return. It's difficult now to, because we can't honestly say that all the mortality that we observed was due to surgery. Some may have died naturally, some of the very individuals that were tagged. So that'll be determined later. And so it can also be applied to gain an understanding of which fish, which sex or which length is more susceptible to post-surgery mortality. So if you're looking at which fit, the healthy ones you want to sample for a study, you can say, smaller males showed a slight bias, they died X amount at the time, where the larger males died X amount of time. So even though it's not significant, we may want to avoid smaller males. And we also use this data to adjust the total number of walleye that are within the lake system following the study. Because these walleye are going to be used for when the Ballville Dam is removed. So they want to make sure there's not just 10 unaccounted for or 20 unaccounted for. I would just like to give a thank you to Stone Laboratory, the associated partners, and Dr. Van de Goot for a fantastic and informative experience. In this case, they are going to, because I read in the proposal they're going to tag another group, like right before they take out the Ballville Dam. And there's a whole system of these receivers going on in a bunch of different studies. So, yeah, there's multiple tagging events going on in multiple years. Then is there any possible variation that can be accounted for by maybe the population shift towards larger males to determine mortality, greater mortality for smaller males, or the shift towards smaller males? How would you account for that?
What do you mean, like the shift towards?
Well, kind of like how Scott was saying in his experiment, how there was, they were growing faster, but not putting on more weight. So you had different sizes in different years.
Oh, this was that was just the average length of the deceased compared to the average length of the ones that survived. We don't know if those are, the proportions in the population of that size. That's completely different. We're just looking at it in a form of kind of unnatural mortality. So it's hard to say in a natural setting whether those smaller males would be favored. We're just looking at, if
you cut them open, jab something in, which ones fare better?
So with your fish that were, you suspected were perhaps dead, and maybe didn't make it out into the bay, can I throw another idea out to you to consider? What if they were in there and they decided to hang in there and continue spawning?
That's, yeah, we were talking about that earlier, and that could be an occasional possibility, but...
Two, right?
There were two, well, because they carried it out for a long enough time that the ones that were up there spawning, or at least a majority, again, you might get the one or two freaks that decide to stay up there, but...
Yeah, you know, I mean, were they males?
There was a mix. For the ones that appeared upstream and then downstream?
Right, just the ones that you thought maybe were bobbers sitting in the...
Oh, within the bay?
Yeah.
Those, I can't remember, frankly.
I just decided, because if they were both males, I would guess that they just decided to stay in there and continue spawning.
We did notice that the males arrived later, so we were trying to account for that.
That's
If it was a male, you know, it appeared later. We gave it a bit more leniency than if it was a female and had kind of an odd pattern. Because the females typically, you know, just go up, drop, and go.
That's right.
You know, I'll get that to you later, though.
Thank you.
You know, just a very basic question, with tagging mortality, what you're seeing, it would appear that the mortality associated with this process is not very significant. Is that how you feel, or how do you feel about this?
Well, right now we have the 23% possible tagging mortality. The other stuff was just breaking down, see if it was associated with length and release, see if there's any other stuff. But 23%, assuming a low natural mortality, that's the thing, we have to know the natural mortality first. Let's just assume for the second that it's pretty low if they're moving out. Then that's 23% of your fish is pretty significant.
Very good.
Hey, well, I'd like to introduce Blair Perry, who Jen and I had the pleasure of having this field assistant for the last 5 weeks. As you could know, this surprised many of you because you've spent a lot of time around me and Blair and Jen, that Blair and I are very different. I like school bars, referencing being a creepy serial killer. I started thinking about that. He was really quiet the first time I met him, and I was like, okay, well, Blair's a serial killer, then I'm like the annoying co-ed who wants to die in the first 2 minutes of a horror movie. We surprisingly got along really well and like had a blast together. We both like names and trailer park boys and science. And I was really, really pleased with how well Blair worked within our system and how excited he got about the science and not just being able to handle it. Obviously, like it's most of us, but I was most impressed with his writing skills that were really amazing for his level of process. I'm really looking forward to keeping him in on my project and doing some stuff with me probably, and hopefully presenting as well. So, without further ado, Blair's going to present his results from the BB bar.
Thank you, Lindsay, for the introduction. I think my mom is watching, so she will ask about the serial killer. But anyways, so my project looked at neonatal growth patterns in the Lake Erie water snake. So I'll get right into it. So a quick overview, I'll do an intro to the water snakes, prior neonatal growth research, project information or introduction to my actual project, the results, further research, and outreach. So the Lake Erie water snake, as most of you are probably somewhat familiar with, is a non-venomous live-bearing snake that is endemic to the islands of western Lake Erie. It's a very constricted range for a vertebrate population of only about 40 kilometers in diameter. As Scott mentioned earlier, their diet now consists mainly of the round goby, which is a shift from formerly featuring native fish and amphibians, but as he mentioned, they are almost entirely on the round gobies now, which actually help them bounce back from their threatened status, which I will discuss in a minute. And their active season is April through October, and they give birth in September. So they were placed under federal protection as a threatened species in 1999 due to habitat loss, human persecution, and the already restricted geographic range. And through extensive outreach and conservation efforts, mainly by Kristen Sanford, they were removed from the endangered species list in 2011. They are still considered endangered in Canada, however. And with the delisting was a federally mandated post-delisting monitoring plan, which involves an ongoing prey consumption study, and a population, just general population monitoring, which is where the neonatal growth and survival studies come into play. So these are the guys that I was working with specifically. The 2014 neonates were the snakes that were born last fall, 2013. Just a quick couple bits of information. The average snout vent length, which is a standard length for snakes, which I will go over soon, was about 188 millimeters at birth, and they have a very low survival rate of about 18% survival. So that is why it's important to study their development and growth to try to figure out what is factoring into that survival rate and how it can be factored into conservation efforts. So some previous studies, I won't go too in depth, but previous studies with neonate snakes have found that in general, larger size and faster growth rate is indicative of higher survival later on in life. This viper species here, it was also found that energy was better allocated to increasing the snout vent length versus mass, so longer was better. Garter snakes here, larger neonates survival species, or higher survival in a natricine species, which is also the classification that water snakes fall into, so very closely related life histories there. And then for this python, they actually found that just high amounts of growth early in life can increase lifetime fitness. So my project specifically was comparing the neonatal growth between South Bass Island and North Bass Island. So South Bass is a pretty large island, about 1,600 acres, a fairly large for an island, 140 permanent residents about, but as you know, it is very high tourism in the summer, so that number is much higher than that effectively during the summer months, which is also when the snakes are active most. And then North Bass Island is about half the size of South Bass and has a very small human population of about 24 permanent residents and does not have nearly as much of a tourism scene as South Bass Island. So some observations that inspired this project was the smaller human population on North Bass led us to believe that there should be it suggests a larger predatory bird presence, as previous studies have shown. So if there is this larger predator presence, that would enact stronger predation pressure on the snakes, which would then therefore bring stronger selection pressures acting on these North Bass Island snakes, both adults and neonates. So the larger snakes would be more likely to survive predation and reproduce, which in the long run, which would produce larger neonates. So in the long run, we would probably see a shift towards larger neonates on North Bass Island, which is actually what we were studying. So hypothesis, North Bass Island neonates are larger and grow faster than South Bass Island neonates due to lower human presence and subsequently higher predator presence. So a previous REU actually looked at the predator densities on North Bass Island, and things you need to pay attention to here is the highest density of great blue herons was found on North Bass, which is the primary predator of the Lake Erie water snake, and also the lowest human density. So that benefits or supports what we were thinking so far. So our study sites, we had one on North Bass, which was the South Shore, which is a pretty, follows a road along the shore, but there's not much human development along the shoreline there, so it's mostly just large rocks and a little bit of a beach. And then on South Bass, we had East Point, which is a nature preserve that was established partially to help the conservation of the Lake Erie water snake a few years ago. And also South Bass Island State Park, which is a very high human traffic area. They have camping, public beach, boat launch, so there's always lots of people around there. This is, I do not have pictures of North Bass, unfortunately, but this is some pictures of South Bass State Park. So you can see that there's you can see the kayaks, you can see the boat launch, there's camping up this way, there's cabins. I'm standing on the boat launch here. So even with all the human activity around, there's still a lot of snakes. These rocks are always full of large adult snakes, and these rubber mats, which are used to launch boats, often have lots of baby snakes under them, which I'm special interest in. Most of these study sites are large rocky shorelines, which were built up to prevent erosion of the shore. And the snakes love it because they can come out in the sun on the rocks and then have a quick escape route when they see a bird or a snake researcher coming for them. So methods were actually quite simple compared to a lot of the studies we've seen so far. I spent the majority of my time outside in the field, which was really nice, instead of in the lab. And the basis of it is running, grabbing, and bagging snakes, as many as you can find. And then once we have a decent amount gathered, we process the snakes, which involves taking measurements of snout vent length, mass, pit tagging. We check for pit tags. If they do not have a pit tag, we insert one just under the skin, which gives the individual snake a specific ID, so we can compare that later if we recapture it. Check for prey, which is part of the prey consumption study. If they have prey, we make them barf, which is always fun. And then morph, which is just the color pattern of the snake. So snout vent length, quickly, is a standard measure for snakes. It involves the snout to measure from the snout to the vent, which is where the reproduction and waste removal happens. They do this instead of snout to the end of the tail because the tail often is lost partially. A lot of the snakes we find have little stubby tails from predators or other accidents. This is a more consistent measure for the snakes. And also the pit tagging is valuable to us because if we have an individual snake that we can individually ID, if we recapture it later on, we can compare the individual growth of that specific snake instead of just trying to estimate it through the collection of general population of snakes. So just to reiterate the hypothesis real quick, the North Bass Island neonates are larger and grow faster than the South Bass Island neonates due to human presence and higher predator presence. So this is all of the data we collected this spring and summer. We did some tests on it and found that there seemed to be no significant difference in growth between the North Bass Island and South Bass Island snakes. Although this data was a little hard to try to get a visual representation of what was going on, so we made these box plots out of it. Quickly, we are missing a chunk of data for North Bass Island when we are unable to get up there to sample, so that is an unfortunate thing. But interesting thing about this view is that North Bass Island, we split the time period that we collected into 4 16-period days and made these box plots of it. So North Bass Island is in blue, and if you look, the mean of North Bass Island is normally, or actually in all of these cases, slightly above the mean of the South Bass Island snakes. And with the addition of this outlier here, the largest snake found in each period was from North Bass Island. So this is making us think that there might still be some sort of a trend here that the scope of my project was unable to ascertain, so we are planning on continuing to look into this. But since this wasn't quite as conclusive as we hoped, we decided to go back to the fall 2013 data which we had collected, which was representative of the snake's size right after birth. And when we did that, we did find a significant difference. The North Bass Island snakes were about 10 millimeters larger on average than the South Bass Island snakes. So, this is a quick slide for fun. 2 of the neonates that we tagged were we recaptured later on, which is extremely rare. As you can see, 2 out of the 80 snakes we tagged on North Bass Island were recaptured. Quickly, I forgot to mention, North Bass is the only island that we pit tag the neonates on because we assume a slightly larger recapture rate than on South Bass when there's a lot more area for them to disperse and lots more human-related death associated. But with just these 2, these are their names, the nice names we gave them. Interestingly, you can see how much they are growing in a fairly short period of time. I mean, 16 days, 10% increase in length, and 32% increase in mass. So they are definitely growing severely during this time period. So why the difference between the fall 2013, which was a significant difference, and 14, which was an insignificant difference? We think that, so they are being born larger, but they're not necessarily growing any faster. So the selection pressures based on, or enacted by the larger predator pressures, should result in bigger females, which we have data backing up as well. And the bigger females are physically able to produce bigger offspring. So that makes sense that if the selection is driving towards larger females, then the offspring would as well be driven to be larger as well because the females are able to give birth to larger offspring. But as we are seeing right now, it does not appear that the growth rate is affected by this predation pressure. which is possibly likely that that is a much harder thing for selection to act upon because, I mean, with the bigger females surviving and reproducing, that's fairly simple in terms of selection and that the smaller snakes are getting picked off quicker, leading to the bigger ones surviving and reproducing. But the growth rate is a much probably more challenging thing to be affected by this selection by predators. So some problems and improvements with our experiment. It was a pretty small sample size. We were limited to one trip to, well, I was here at least, one trip to North Bass Island a week. And one of those weeks we only found 2 neonates. So if I could improve, I would try to sample a lot more during the week and also try to sample for a longer time period in order to get a larger sample size by which to go with. The low recapture rate, as I mentioned, it's extremely rare to recapture neonates, like 2 out of 80 on North Bass. This isn't something that we can necessarily improve because of the high mortality rate, and also it's just sometimes you find a lot of snakes, sometimes you don't. But if we could find more, then that would provide us with the individual changes in growth changes in growth for individual snakes, which would provide more insight into what's actually happening. And additionally, since we did see that there could be some sort of trend that's really close to between North Bass and South Bass, but might be a little larger in North Bass, factoring in some other measure of body conditions such as mass would give us a more comprehensive view of neonate size in order to evaluate the hypothesis a little more detailed. But due to lack of time in the study suggesting that the SVL length measurements were more indicative of survival, led us to just go with the SVL measurements for this project. So some further research. This is just a piece in the puzzle of the long continuous Lake Erie water snake research. So it doesn't necessarily seem very extraordinary, these results, but hopefully they will serve as at least a basis for later studies, or Lizzie is actually doing feeding and growth experiments in the Lake Erie water snakes this fall. So this might prove somewhat relevant for her research, and also somewhere else she could focus her research in the future. We also have a large database of all of the captured snakes for the past 30 years or so. So if time would allow, going back in and looking at neonate measurements in previous years and seeing if we see similar or different or any trend at all in the growth of those snakes. And additionally, just continuing outreach to make sure that the populations are not affected as negatively as they were in the past by human populations. So quickly before I finish, a couple more things I did while I was up here that I'm considering outreach. I worked on the Respect the Snake website, which Kristen made in order to provide the public with a quick place to get educated on the Lake Erie water snake, kind of bust some of the myths that there are out there for it. So worked on updating some of the information and updating the user interface a little bit. It's not live yet, but hopefully in the next few weeks it will be. Additionally, there was a Canadian film crew here last week, which some of you probably saw running around, and they're filming a documentary series on Lake Erie wildlife. So I don't think it'll be out until January of next year, but we had a really great time, and they seem to have a really good vision and are focusing more on the recovery and the beauty of the snakes rather than the biting and the pooping and all that stuff that previous TV shows have focused on. So I want to thank Dr. Kristen Stanford, Elizabeth Mack, and Jennifer Beck for all of their work closely with me in the past 5 weeks and for guiding me through this. This is my first research experience, so I probably needed quite a bit of guidance at first, but I think I caught on eventually. So I appreciate them for all of their efforts and bearing with me. Dr. Reuter, Friends of Stone Lab, and Dr. Winslow for everything that they've done to make this happen, especially Friends of Stone Lab. I would not have been able to do this if it was not for the scholarship that they were responsible for. My wonderful teacher, NTA, for my field zoology class, who bared with me running and looking for snakes while we were supposed to be collecting butterflies. Dr. Justin Chaffin for everything he's done to organize the REU program, all the Stone Lab staff, and also the snakes for always being cooperative and friendly during our collection time. Any questions? Yes.
Do you think people flipping up the maps on islands or anything to like make a study bias?
Possibly. We actually even lost some maps on South Bass throughout the time of the study, which was frustrating. But a a lot of the babies we find under rocks. Sometimes we find them under the mats, but more consistently we find them under rocks along the shoreline. So in general, I mean, obviously people are constantly messing with those mats when they're not on locations where we can easily monitor. So that's definitely a factor, but hopefully it will not have any significant effects on the study overall. Yeah.
So your hypothesis and your selected pressure stuff seems really sound. But I was wondering, do you know how much connectivity there might be between South Bass populations and North Bass ones, and maybe that be the reason why you're not seeing them?
I mean, snakes going back and forth between? I'm not sure. I would guess that it wouldn't be very significant given the distance. I mean, if they went to Middle Bass and then came over here, I don't think there's much direct going back and forth just given how large that distance is. But it's definitely possible. I mean, it's neat because all the islands are slightly different conditions, which provides a lot of, for studies like this, when you're comparing the populations on each island out of the entire general population. But the fact that they do go in between definitely can skew that. Sarah?
I'm not sure if there are any local populations of the northern water subspecies, but could breeding differences, gene flow between that subspecies affect growth rate?
Definitely. I didn't go into this too much earlier, but when we were talking about the morph, the unbanded water snake is the typical Lake Erie water snake, pure Lake Erie water snake, but I would say close to at least half of the snakes we find are actually heavily banded, which means that there's been interbreeding with the northern water snake somewhat previously in their generation, or their, so it definitely have an effect. I don't know necessarily what effect that would have, but that is definitely, there's constant interbreeding between, so exchange between the populations.
Like maybe islands that are closer to where the northern water snake populations are might have higher levels of gene flow, or...
It's definitely possible. Something that I did not look into, but I'm sure has been looked into in the past and can be applied to this in the future, I'm sure.
Going on to that question, would it be a possibility that or the frequency of the different color morphs between the islands, like C is the one, less human population, have a typical plain coloration, blend into the rocks, one closer to humans, have a banded population, just like how the context is with predators and such, that's something that they're selecting for.
Yeah, well, quickly about the morphs, the reason that the Lake Erie water snakes are technically unbanded, the pure ones, is the northern water snakes are banded because they spend a lot of time in grasses and foresty tree areas, so that provides better camouflage there. But these Lake Erie water snakes are often on rocks or swimming in open water, where the plain solid color is often more beneficial in terms of hiding from predators and stuff. So they're definitely, I don't know if have there been studies with the proportion of morphs on different islands?
Yes.
Okay, so there have been studies with that. I did not apply that into mine because I think also the morphs can change the maturing of the snakes from neonate to adult.
With North Bass actually has a higher percentage of unbanded than our near mainland islands because of the higher rate of
Anything else? Yeah.
Among the DNA, the still brown gobies that you
For them, it was a lot of shiners, occasionally gobies, smaller gobies, but I think I didn't, for this project, I didn't actually look specifically at the proportion of which prey was the neonates I collected, but saw a lot of shiners, some small gobies, because they're gape limited, so with their small size, there's probably more of the shiners that stay around that size around than the baby gobies at the time. So.
Thank you very much. We have one more speaker.
Last, not least, we have Rachel. These guys are coming to us from North Central College, which is in Naperville, fellow refugee from the Central Time Zone.
Actually, football and baseball, I'm sorry.
We're waiting for hockey season. We've abandoned our inaptly named Great Lake of our own, from the Lake Erie to play with birds and harass us. Without further ado,
You still haven't caught me yet.
I'm
unfortunate distinction of being the last speaker, and you guys are all very tired, not happy about being way later than we usually go, so I'll try to make this quick and have you get the heck out of here. So I'll start with what we've been working on. Like Dr. Marshall said, we've been working on birds with the Lake Erie Islands, dealing with suitability of the Lake Erie Islands as bird habitats, breeding habitats. So just a little background on the islands. Lake Erie Islands, as well as other islands from previous studies, have shown that small islands near the coastline on a large body of water are crucial stopovers for several species of migratory birds. What we don't know is how well these islands stack up as breeding habitats. So we have a wide variety of different habitats on these islands, anywhere from human-dominated areas like residential places all the way down to natural preserves. So we initially had some criteria that we wanted to use. The first one is nesting success. We wanted to measure how many hatchlings were being born, but unfortunately we started this project a little late in the nesting cycle. So most of the birds had fledged by this time and we couldn't really judge by how well they were breeding. The second one is survival probability analysis by measuring how many recaptures we got during the study, but unfortunately we did not get that many recaptures. So that option is also out. So we've got two hypotheses that we're working with on this project. First one is that birds will act to maximize fitness in the best habitat they can. And the second one is that older birds will dominate younger birds in the better habitat. Therefore, if these hypotheses are supported, we will consequently see more older adults, more experience, more territory. You'll also see more offspring consequently. And we will also see greater health overall in the adults in better habitat. So, a little background on the islands. We use 4 islands for banding. We banded on this island around the cottages and solar panels. We also used Chief Preserve and Bayview Office on South Bass, East Point Preserve on Middle Bass, and the vineyards and fields of North Bass. So we did our banding from June 24th, July 18th in the mornings. We used both REU time as well as class time for the evolution class band for this project. We had 10 mist nests that we used every time we went to a site, and we had a specific set of measurements that we took for each bird: wing chord, fat, age, sex, and weight. We also noted any damage that we observed on the birds when we were working with them, such as where they had a deformed beak, where they got any cuts while they were in the net, if they had any pox on their feet, which I'll get to later, or if they had wing stress, having difficulty flying away after we banded them. We also took extra measurements for the tarsus for blackbirds as part of our condition index. And speaking of analysis, we used 3 different types for this project. First, we used chi-square with comparing 2 different sets of age ratios. The first is measuring the ratio of older adult blackbirds to younger adult blackbirds. And the chi-square is measuring the significance of that difference in ratios between. And we did the same for the total number of adults versus the number of hatch years for blackbirds and also for robins. The next thing we used was a condition index to measure the overall health of the birds in each habitat. And to do that, we ran a regression of mass as a function of tarsus length, and we took from that averages to compare on a t-test, and from the t-test we used another p-value to determine the significance of differences in health between the birds between the 2 populations. And as I said before, we've been seeing a little bit of pox appearing on some of the birds' feet, so we also graphed the frequency of pox over the years of the study. And pretty birds, because we can't do a presentation on birds not showing actual pictures of birds. So this is the one that's most common. This is our red-winged blackbird that we so love. This is our robin, that's the second most common bird. We also got cardinals, a lot of hatch
pitchier yellow warblers, and orioles. A few species of swallows, my favorites. A couple species of vireos, and a few species of flycatchers. So, this is the total number of species that we got this year. It's pretty on par with what we've got over the other 3 or so years in the study. And this is how it breaks down between all the islands. We obviously caught fewer this year because this is just 1 year compared to the other 3 years, but the pattern overall is still the same. We got the most species of birds on Middle Bass and North Bass, Gibraltar not too far behind, and that's pretty much been the pattern for all the previous years. So for our chi-square test, just a little review as to how this works. What we're doing is we're comparing 2 different age groups of birds. The older birds that are labeled ASY and the younger birds that are labeled SY. And what we said in our hypothesis is that we want more ASY birds. They're more experienced, they're going to have a better chance of getting territory, and we want higher ratios of those birds in the territory. So what the chi-square does is it measures how significant the difference is between the ratio of older to younger on human habitats and the ratio of older to younger on preserves. And for our 2014 male blackbirds, we did not find a significant difference. It's a critical value of 0.05. And same pattern with all years blackbird males. Ratios are not that different, still not significant. And we see a similar pattern with the 2014 females. Now, this result is significant. This is female blackbirds spanning all years, and it shows that there are a lot more older female blackbirds in the human habitat as opposed to preserves. The ratio is much more evenly spaced between older and younger in the preserves. So from this, we can clearly see that female blackbirds across all the years prefer the human habitat. And consequently, there's more females, we're probably going to see a significant result in the hatchier blackbirds. We did not see a significant result this year, but we did see a significant result across all years. With the females that we just saw, there are a lot more hatchiers in the human habitat than there are in the preserve. So we're seeing a pattern here. We're seeing a trend with human habitat being favored. We also did the same thing for robins. Now for robins, both this year and across all years, we did not see a significant difference. Ratios here and here, they're pretty even. But we do see that robins do slightly favor the preserve for their hatch years. And we also did the t-test, which means that we're taking the regression of mass over tarsus length, and we're using that as an indicator of health. So we ran a t-test, we took the residuals from regression, ran a t-test, compared the averages, and we're not really seeing a significant difference in the males or in the females. Pretty same patterns throughout the study, not really much difference in health between all the years and this year. Now this is our pox, the interesting little thing that I mentioned earlier. It forms as these swollen, dry, scaly lesions that we can see with some waves on here. Oh well. Yeah, there it is. So it's very dry, it's very cracked. Poor guy. That's what we're seeing on a few of the birds that we've caught. It's fairly uncommon. It's only the peaks that we've seen last year only got up to just under 3%. But it's still an interesting form of disease that we want to keep an eye on, see how it changes over the years. So to recap our hypotheses, we got 2 working hypotheses. One is that birds will have the best fitness and best habitat, and 2, older birds will dominate younger birds in the best habitat. And if we see these hypotheses supported, we will consequently see more older adults, more hatchier birds, and the adults will be in overall better health. So, summary of our results, we did not really see that many significant differences between the human populations and the preserves. The analysis that we did, the age ratios were pretty even, health conditions were pretty even. We did see a couple patterns, though. The hatchier blackbirds did favor the human-dominated habitat, and the robins did show a slight favor of preserved habitat. We
we did not see that many significant differences overall. This was our, the P is greater than 0.5, it's not significant. Our two exceptions were the female and the hatchier blackbirds from our all years analysis. So there is a bit of hack going on there. But keep in mind that this analysis was only used for the two most common species we had in our study. It does not account for all the other species that we encountered. And, like I said, keep an eye on the pox. It's not very prevalent right now, but it could go either way in the near future. So, should we invest in these preserves? Definitely. I mean, there are a lot of different birds that are dependent on these. However, it does not necessarily mean that the blackbirds will use the preserves. The blackbirds, overall, except for some females and hatchiers, didn't really show a preference. However, we got to remember that there is ample habitat around this area for these birds, both on the islands and in the mainland. They don't really have a major limit in their habitat choices here. Now, let's not forget that blackbirds especially, they're a little aggressive, but the birds in general, all of them could very well be competing for this habitat, so that could also have affected our results. So, in conclusion, most of the differences we saw between the human habitat and the preserve habitat were not significant. Our only two exceptions were all years female blackbirds and all years hatchier blackbirds. We do want to keep our preserves up and running for all the general species, because other species vary in needs. But the blackbirds might not necessarily choose one habitat over the other. So in the future, we want to take our blackbird methods and we want to apply them to other species that we found to be more common in our study, like all the hatchier warblers and orioles that we found. That'll give us a better idea of where those species like to go, how they're using the habitat, what other species need the habitat. And we also want to get more recaptures than we did this year. We got very few this year, and we want to get more next year because we want to run a survival analysis. And finally, we also want to track the pox. It's a disease that we've only just seen in the past few years, but we want to keep an eye on it and make sure it doesn't get too serious. So, with that, I want to thank some distinguished people for helping out with this, mainly Stone Laboratory, Friends of Stone Lab, Ohio Sea Grant for funding this research. It would not be possible without you. Also, I'd like to thank everyone back at my school, North Central College, for their support. I'd like to thank Dr. Marshall for being a great advisor, all his advice and insights, dealing with all our mishaps out in the field and all our little adventures. And Dr. Jeff Reuter and Justin Chafin for running this REU program, giving us all this opportunity. I'd like to thank my classmates, because we got a good chunk of our data from the time we spent banding in class. Also Tom Bartlett and Lisa Broll, two other banders that met us on North Bass, gave their support. Also the boat captains, Art and Russ, for taking us to and from the islands. And last but not least, the birds, for having to put up with two big scary humans taking all these crazy measurements. So with that, I'll take your questions.
So I know I've heard you talk about it in like our elevator discussion, and I think I kind of know, but I'm not really sure what it's.
A tarsi, well, with birds, allow me to have a little visual aid here. With birds, the knee part is actually, you can't really see it most of the time. It's covered by feathers. The ankle part is actually what you see, the narrow spindly part of the leg. So the tarsus is measuring from the joint where all the toes are to the base of the ankle. So that is, that's what the tarsus is. We're measuring from joint to joint. Yep.
So you talked about pox and that we need to keep an eye on pox and the scaly lesion for feet, but what does it do?
We suspect that it's caused by a type of mite. I believe the proper name is Nimidocoptes. It's pretty much the avian equivalent of mange, like the mites that we have in mammals that cause acrostic mange species. And they basically, they burrow into the skin, and it causes lesions a little similar to, like warty type lesions. So, yeah.
Is it painful at all, or is it?
It's definitely uncomfortable for the birds. We had of the 3 or so birds we had with POTS, at least one of them had feet that were all cracked and bleeding, and that's because of the dry lesions. We're not sure how it affects the bird's overall survival right now. That's something we definitely want to explore in the future. Like, possibly, if we have the resources in the future, maybe take a few samples, maybe methanol solution, see if we can test some of the material from the skin, see where the disease might be coming from. So, yep.
Was there any noticeable increase in like aggression when you were handling them?
The birds varied.
Yeah.
The birds varied a
I
lot.
mean, the blackbirds are pretty mean.
Yes, they are very aggressive. And I did get bit by a cardinal one time, which hurts like heck.
Oh, yeah.
And, but some of them were more docile. The flycatchers, the swallows, goldfinches, warblers, they were pretty docile. But blackbirds, even some larger birds that we caught that we didn't have a band size for and had to let go. We caught a flicker, we caught a few grackles. They're a little mad.
Do you know the different kinds of birds that you call it fox, or the three like individuals?
I'm pretty sure they were all blackbirds. We only caught 3 this year.
What was the frequency in catches of 17?
I don't believe we stole across any
There's some very close misses.
Do we need to increase our capture probability?
I think we need more tags.
So Rachel, the frequency you're seeing of the fox on the island, how does that compare with the frequency on the mainland? You know?
We haven't studied the frequency on the mainland yet. I'm not really sure what that would be.
This year wasn't last time. Hopefully it's going down.
Very good.
Well, again, these were excellent presentations. Congratulations to all of you. You can sort of breathe a sigh of relief now. You really did a very nice job on these. And for most of you, you had almost zero experience in these areas before you started. So you clearly want to recognize the advisors. You guys also did and I really want to encourage you not to stop here now. I know that you really ought to think about, you're so close to, those presentations. The presentations you made today, these are good enough to do at the Ohio Academy of Science, at AGRA. Remember also the lady that was here early in the year, and she talked about the opportunities to present over Erie, Pennsylvania, with the Pennsylvania group. That's another good spot, and they'd love to have you go, and we want to help you get there, so keep that in mind. Chris, do you have anything you want to add?
Actually, to piggyback off of that, I know it's a short turnaround, but there's an August 1st deadline for a undergrad presentation on OSU's campus. That's the deadline for the abstract. So if you're an OSU student and you want to present next semester, and I can get that details to your supervisor.
Good. Justin, any final comments?
I thought all the presentations were really good. Oh, yeah, so you need to turn in an abstract or an extended report to a supervisor or directly to me by Saturday in a PDF form.
So by Saturday, you're going to have your abstract ready that you can give to Ohio State to submit it down as part of the demo. Keep that in mind. Dr. Weisenberger, our Senior Associate Vice President for Research, any comments from you? You've seen these for quite a few years now.
Yeah, this is hands down my favorite day to come up to Stone Lab every year because I'm always so impressed with the quality of the work you guys do. And also really jealous that you've gotten to be up here for the last 5 weeks doing this work. It's the best possible summer experience you can have. And it will change your life in all kinds of unexpected ways. some of you may be thinking differently about a career path you might take now. Yeah, I see the heads nodding. For some people, they don't know necessarily that they're going to love research until they start doing it, and then it kind of sucks you in, and then you're sort of hooked for life. So we hope that we've captured all of you, and that you will go on and have outstanding scientific careers. And as Jeff said, StoneLab is here to help you. So take advantage of those opportunities to present, to work with your advisors to publish the data, to ask for recommendations when it's time to look for jobs or grad school. All of that, this experience this summer can be totally critical in all of those endeavors that you're going to go into going forward. So thank you, because I had a fabulous evening. I learned a ton tonight. So thank you all very much.
I owe you all a trip, and sometime I don't think I'm ever going to catch up with you, but normally we go to the Dairy Queen after this, and we have to be there by 10 o'clock. We're not going to make it. But these were, Jeff, 8 o'clock? Yeah, there's possible.
They did say the dining hall was going to stay open until the end.
So we'll go get for the ice cream there, but again, very, very good job. Congratulations. Hey, Hunter.